afternoon and welcome back, everyone. Just a brief introduction of Dr. Hamid Merchant Saab. Dr. Merchant is the subject lead in pharmacy and the course leader for postgraduate suite of pharmacy programs at the University of Huddersfield. Sorry. Dr. Hamid is also an adjunct professor at the Health Services Academy, Government of Pakistan. He has over 18 years of experience in the pharmaceutical research and development, both from the industry and the academia. His expertise includes novel formulation design, biopharmaceutics, pharmacokinetics, bioavailability, and bioequivalence. His clinical knowledge and expertise in pharmaceutical formulation helps in understanding the clinical and therapeutic principles underpinning the science of dosage form, design, and develop innovative solutions to improve the delivery and efficacy of medicines. Dr. Merchant has also contributed significantly to the evidence synthesis in COVID-19 to help safe use of medicines amid pandemic. He has also proposed an early therapeutic intervention strategy to help prevent the disease progression and hospitalization. Before joining Huddersfield University, Dr. Merchant has been associated with the University College of London, which is known as UCL, where he investigated various technologies for drug delivery into gastrointestinal tract and offered technical support and expert advice to collaborators and clients from academia and industry on various formulation and drug delivery aspects. One of his post-doc assignments at the UCL was to develop a novel formulation for colonic delivery of statins. During his time at UCL, he has also developed novel in vitro solutions for the assessment of pharmaceutical and nutraceutical products in physiologically relevant conditions of the gut that helped a number of companies in formulation optimization and candidate selection for clinical trials. One of the technologies that he optimized has recently passed phase 3 clinical trials for the inflammatory bowel disease and is due for launch in the UK and elsewhere in Europe. Dr. Hamid was associated with Abbott Laboratories Limited from December 2002 to November 2007 and served in various capacities such as quality, formulation, new product development, continuous improvement, and project management. He is part of editorial board for various scientific journals and is also a deputy editor-in-chief for the British Journal of Pharmacy. Dr. Hamid is a fellow of the Higher Education Academy, member of the Royal Pharmaceutical Society of Great Britain, American Association of Pharmaceutical Scientists, Controlled Release Society, and the Academia of Pharmaceutical Sciences in Great Britain. Dr. Merchant has a pharmacy degree with a master's from the University of Karachi and a PhD in clinical pharmaceutics from the University College London. He also has a PGD in quality management from the NED and a PG Cert in higher education from the Huddersfield. Can I invite him? Dr. Saab? Dr. Saab, we'd like to invite you further and let's take the session ahead.
Thank you very much for the nice introduction, and thank you very much for all of you for staying after the lunch for the talk. Normally it's very difficult to keep the audience after the lunch. So I was talking to Kaisar Saab and saying either everybody would leave after the lunch, or if they were scared to leave because of you, they might fall asleep. So I will try my best to engage you and share some of the knowledge I have acquired over the period of time in pharmaceutical sector. But if you feel like you are tired, or if I'm going too slow or too fast, do give me some feedback. Or if you want to take coffee break in between, please do feel free to let us know. Otherwise I can carry on as soon as I'm concerned. So thank you very much for the PPMA, particularly Kaisar Saab and Saeed Qureshi in the morning who came over to start the session, all the other honorable guests from the PPMA, and all the pharmaceutical industry representatives here. If I've missed anyone, I do apologize, but thank you very much for organizing this session and for all of you to attending it. I think it's one of the important initiatives after COVID-19 to reconvene in a platform like this and have the knowledge sharing experience within the industry. And this knowledge sharing across the industrial platform is so important for the betterment of pharmaceutical industry in Pakistan. So moving on to today's session, first, lots of people ask about Huddersfield. It's not a very well-known place outside United Kingdom. So you might have all know about London. So that's where the London is, right at the bottom. And if you look at the Huddersfield, we are right there on the top. And it's very close to three main cities which you are aware of. So the Leeds on the top, Manchester at the bottom, and Sheffield around the corner. So you're very well connected to all those main cities, and that's where we are based. So I'm based in the University of Huddersfield, where Professor Zahir Babar is also based. So we have a very good team at the Huddersfield University, having staff from discovery, formulations, and clinical practice and public policy like Zahir Babar. This is just a snapshot of our main university reception, but university is very big. It's a multi-faculty university offering 250 programs, undergraduate and postgraduate programs with about 25,000 student population on campus. So it's a proper state university, if you like. It's a public university catering for the local area. Majority of our students are English students around the Yorkshire region, but we do have about 7,000 international students coming from about 150 different nationalities. Right, so today's talk will look at the role of drug development and discovery, as Kesa Saab asked me to talk about it. So to give you a perspective to begin with, I'll talk about the global perspective, that how pharmaceutical industry gets involved into the delivery of healthcare and why it's one of the fundamental pillar in any country's healthcare delivery. So look at the global perspective, and then we'll move on to the specific example of Pakistan as to how we currently form the fundamental part of healthcare delivery in Pakistan, where do we stand, and how we can go further like other countries or developed countries in the world. We will look at the future perspectives, looking at some opportunities where our industry could move on. We have been on this journey for a long time. Pakistan industry has done a great job in supporting medicine supply and equitable access to medication, as Professor Zahir covered in the previous session. But now I think is the time to change. And if we do not change, we will suffer a lot in the coming period of time. We are already late in the game. So I'll talk about some of the perspectives, and some arguments around discovery always comes to the cost, affordability, and people say in the generic pharmaceutical sector, innovations are difficult, discovery is difficult, we cannot be a discovery-based organization. So I'll give some case studies, some examples from my own experience where innovations can happen within the generic pharmaceutical industry without spending millions of dollars on discovery. And I'll give you some very brief overview, some suggestions, not extensive list, but the sky is the limit in terms of your innovative ideas. If you think out of the box, you could always come up with an innovative product within the supply chain of Pakistan and the product needs. So let's start with the global perspective first. And I take this example because we have all been suffered a lot during pandemic, and it was not just Pakistan, all over the world. It affected everybody's life. This is a good example to understand how drug discovery and pharmaceutical industry chips in to support the public health, or we call it in the modern terms global health security. So like every country have a policy to protect the borders and security of the country, law and order of the country, similarly the safety of the nation can also be compromised by the diseases and the pathogens and the pandemic-like situations that we faced. So global health security is one of the top agenda, not only on WHO, but if you look at the United Kingdom, it's one of the top priority of the country to make sure that the health is secured for the British population. So if you go back to the earliest we can in our textbooks knowledge, where we first time learned about the pandemic and how things happened, we never realized that we will see the pandemic in our lifetime. We thought maybe the people were not that efficient at that time, technology wasn't there, pandemic will never come, but it did came. So 1918, Spanish flu, moving on to bird flu, 1997, similar sort of scenario, taking many countries on its coverage, swine flu in 2009 and 10, and then GSK's product, Pandemrix, to encounter the flu preparedness at that time, and there were lots of controversies around pandemics, the vaccine from the GSK at that time, and I was in UK when that was happening back then. So the COVID repeated itself in terms of lots of issues we have seen, and there were lots of lessons actually to learn from the history as how these viruses evolve and how Public Health England then reacted to encounter those. And then we had SARS. Everybody would have heard about SARS, affecting the part of the society. Fortunately, it didn't turn into a pandemic, but it was an endemic, and it did affect Middle East and some of the countries very, very badly. And why the countries like Middle East didn't had the preparedness plan that we had now for COVID. I'll talk about that briefly later in the talk as well. Because it wasn't pandemic, the SARS wasn't tackled like COVID was tackled. And then we had MERS, which was another version of SARS-CoV virus, and because this was affected mainly in the Middle Eastern region, when the virus jumped from bat to pigs in the first stage, and then from pigs it went to the camels, and then Middle East got affected with the MERS virus, which was one of the variant rolling out from there. And then we had the COVID-19, another coronavirus, jumping out from the bats and then rolling from China to across the world. So this is 1918 Spanish flu. This data is from CDC, and this figure shows the pandemic life cycle curve in the United Kingdom, and you can see 3 distinct peaks. These peaks shows the mortality or the deaths per 1,000, and you can see that at that time we also saw multiple peaks, mortalities, and also you could see in the summertime period as well, which is quite awkward to have flu virus spreading out in the society. After 3 distinct peaks, the virus subsided down. At that time we didn't have any vaccines at that time, and the World War was phasing out. It was a very difficult time. It turned into a global pandemic, but the pandemic subsided down. But what happened to the flu virus? It never left us. Okay, the virus is still within us. It is part of the society. So when COVID-19 came in, I jumped on. People were talking about zero COVID, eradicating COVID from society. And I'm like, hang on a minute. Have we not looked at the history? So I wrote at that time in British Medical Journal saying virus is going to stay with us forever. And that was April 2020. Everybody laughed and criticized. And you can look up that article. What I said at that time is still true. So history is important. If you do not learn from your history, you will again going to make some mistakes. This is the flu outbreak data from Public Health England from Euromoma model, and you can see every other year you have either H1N1 coming back into the England or H3N2, and the number of that every year, which we try to control by annual immunization program. So you might have heard about flu vaccine, but flu vaccine is not just a flu vaccine. You look at the variety of flu vaccines we have in England. And how do we personalize vaccines for different age groups? So we don't have just generics or copy of the flu vaccines coming from different companies. They are different technologies. They're made using different antigens. They're made using different formulation strategies to cater for different age groups, for people with different pre-existing conditions. For example, certain vaccines are contraindicated. Some flu vaccines cannot be given to immunocompromised patients, but the other ones can be given. Some can be given to children as young as 6 months. Some cannot be given to anybody below 18 years of age. So there is a variety. Not every vaccine is same. Not every product is developed the same. And not every product has the same antigen. So clinical efficacy and immunology changes as you change the formulation and makes it unique for that particular population. One of the unique vaccine which you would not have heard about in Pakistan context is the Fluence Tetra. This was introduced quite recently by Public Health England. Recently means in last 10 years. And this was a step change in controlling flu in the community settings, in the schools, in the societies for the community spread. And this vaccine was produced by the AstraZeneca, and current version of this vaccine is a quadrivalent vaccine. So in the beginning it wasn't a quadrivalent, but this was a nasal vaccine. So it's been given as a nasal spray formulation. It's non-injectable vaccine given to all school age children without having an injection, going to the doctors or surgeries. The nurses within the school settings deliver that within the schools. And the beauty of that vaccine is it produces immunoglobulin A on the mucosal surfaces of the nose and the throat, which means it provides the protection at the site of entry of the virus, which helps stopping the community spread, which injectable vaccine cannot achieve. This is a live attenuated vaccine, so it provides a very good cellular immunity along with the antibody response, and that has made humongous change in the flu pandemic statistics in the UK since the quadrivalent vaccine came out just 2 years before pandemic. Just quite recent reformulated version from AstraZeneca. And then you have got the classic vaccine designed on chicken eggs, the embryos propagated on the egg system when the virus has been cultivated. Then there were issues about virus manipulating within the egg and changing from the actual strain causing the disease. So the newer technologies like cell-based vaccine technologies, where the virus is now being propagated on the cultured cells instead of a living body like a chicken egg. The third generation vaccines are made using genetic engineering, so they are used to produce the antigens directly by microbiota, for example, E. coli based production. So what do you do is genetically modify the organisms, and the organism produces the antigen of your choice. You isolate it, purify it, and then produce a vaccine. So it's rDNA technology, which is external to the body. So you are not inserting any genes into the human body, but what you are trying to do is modifying a living organism outside in a culture system, which is producing the product for you. You isolate it, purify it, and you use it. And that is the modern vaccines. All the two new products launched just before pandemic, one from Sanofi and the generic version from Seqirus, based on the same formulation. But you can see the age groups are different. So they're not indicated for young people because the lack of clinical trials and safety data in that age group. So this is how you personalize. This is how the industry comes in. You do not just copy products. You start innovating within the sector. Try to come up with, okay, what's wrong with current product? Why there are adverse events in some population? And you then modify the product for that niche market areas. And what is missing in Pakistan is this innovative thinking, rather than just competing for the same product by 5 different companies, or sometimes some molecules are produced by 50 different companies. And that's where ethical marketing comes in as well. It becomes difficult to survive in that competitive, crowded market where you've got everybody selling the same stuff. So you need to come up with innovative ideas within the same product and then target those niche market areas. And flu is a very good case where you can learn how formulations and changing the variety of the products could cater different markets. And every product sells. So Public Health England buy all these 5 products for the annual campaign. So they don't buy one product, they buy all 5, and then they prioritize the age group and pre-existing conditions accordingly. So this was the MERS, and it's an interesting to see just before pandemic came in 2020, MERS was still there. It never died. It didn't vanish from the society. And the group of scientists in Oxford University who developed the first COVID vaccines in terms of AstraZeneca being producing it, the same group produced the vaccine for MERS using the same platform technology. And this was the reason that they were able to produce the vaccine for COVID very quickly, because the same adenovector platform was used for MERS. So what they did was basically modified the adenovector with a COVID genome instead of MERS genome, and vaccine was ready within weeks. All the time took for the vaccine was for the safety and clinical trials, but the actual production was just in weeks. It was just done. But why we didn't have the vaccine for MERS? Because it was a localized problem. Vaccine was produced, and it never completed its phase 3 trials, because by the time phase 3 trials were running, everybody in Middle East had the virus and there were no people left to try on. And then subsequently it was not a health priority. So the governments in UAE and Saudi Arabia didn't fund the vaccine program afterwards. So the vaccine never rolled out. to the society. And that's the difference. When the nations takes control, when the nations takes priority on their public health, when they plan the global health security of their population, this situation wouldn't happen. But we learn from the benefit, from the knowledge, the time is spent on developing MERS in terms of getting this SARS-CoV-2 ready in a matter of weeks, if you like. So if you look at the coronaviruses, they're not something new. They've gone back a long time. Some coronaviruses are still, were still part of the human population as people were living with it. For example, if you look at the Texas health data, they had 4 coronaviruses reported every year for about last 10 years, even before pandemic. And they cause mild flu-like situation, and it was never being on the radar as a global health, security issue. And the first one which caused an issue was the SARS virus, which was very lethal compared to the other 4 viruses. And then the MERS, and then the SARS-CoV-2. So the last 3 caused a big threat to the global health security, but the other ones were like a seasonal cold infection. Now the issue in Pakistan is that anybody who has got a viral infection, we would say, oh, it's a cold or it's a flu. Not every viral infection in the respiratory tract is a cold virus. Cold is a virus, common cold virus, and flu is a different virus, and coronavirus is top 4, also cause cold-like sickness. And there are different viruses, and there are different diseases, and they cannot be treated with the same condition. But normally respiratory viruses, the first line of treatment is a symptomatic care, which is why you do not try to differentiate and differential diagnose that which kind of virus it is. So in UK, what we try to do is if a patient is hospitalized with a respiratory virus and the patient is severely ill, then they would do a differential diagnosis. They would take a swab sample, they do the PCR. So PCR is not new. We do it for 20, 30 years on hospitalized patient with respiratory illness. Because sometimes it's respiratory syncytial virus, RSV, which is very nasty. So you need to know. As soon as the patient comes into the hospital, we need to know which virus it is. Okay? So you heard about PCR testing during COVID-19, but this is not something new. We've been doing this in England for a long time, to differential diagnose. But we don't do it in community settings. We do not bother, because majority of people will just recover. So in terms of the COVID vaccines, if you look at the formulations, if you look at the varieties being developed in a very short period of time, the first one you would have heard in Pakistan was the Sinopharm vaccine, which was a whole inactivated virus. So very classic vaccine formulation, and the adjuvant was also alum. So it wasn't anything novel, very classic formulation used for hundreds of years. And here you go, you've got a full antigen, so your body is exposed to multiple antigenic sites, so your antibody levels might not be same as other vaccines because the antibody testing only picks up the anti-spikes antibodies. When you have inactivated whole virus vaccine, your body produces multiple antigenic response, including cellular immunity. So it's not a good idea to look at the antibody titer when you had this type of vaccines and say, oh, I haven't got a response. We need to understand the clinical immunology, try to see what antibodies are triggered by different antigens and how the immune system responds in terms of overall protection against the virus. And spikes are the worst part of the virus because they change so quickly. And targeting spikes for immune protection is a problem because that's changing very quickly. But the nucleocapsid protein, which is under the spike on the shell, doesn't change very quickly. It takes at least 12 to 18 months for nucleocapsid to go into mutation. And that's a very good strength. So if you have a multiple antigenic vaccine, you have broader protection, if you like, broad spectrum immune protection, if you like. But the trouble with Sinopharm vaccine is that it's because it's an inactivated virus, sometimes body doesn't see it as a threat. And lots of people do not immunogenically respond to the antigen. And that's where you suffer on the efficacy sometimes. So people, elderly people sometimes, people on steroids or immunosuppressants, they would not show very good efficacy. Very classic issue with these vaccines. Nothing new, not just in COVID, it happens with all vaccines. Another vaccine, Sinovac, exactly same as Sinopharm, both coming from China, but this used CN02 strain, and they used the first HB02 strain, the first one to come out. Same formulation, same adjuvant technologies, but a different seed, different strain to provide the coverage within China. Then you had the Covaxin from the Bharat Biotech. Now, what India did was use exactly the same formulation for the Sinopharm. There's nothing different in this one, whole inactivated virus vaccine. But what they did was they used the TLR7 agonist as an adjuvant instead of the traditional LM-based adjuvant system. That was a clever move. This was a patented product, so they licensed it from the company, and that changed the efficacy profile of the classic inactivated vaccine. Because what this adjuvant does, it activates your immune system. So your antibody response is 10 times higher with the same dose of antigen. And that was launched without even completing phase 3 completely in India at the time, criticized a lot. But if you look at the science, if you look at the immunology and the design, very clever. But they copied it from somebody else. So this was originally designed by a French vaccine manufacturer who is based in Scotland and was heavily funded by British government, about 90 million pounds at that time, to produce a vaccine. They do prepare some vaccines for things like Japanese encephalitis, and it's Valneva is the name of the company, and they started this one. Then they could never finish the product. They were running low in their profile, but Bharat Biotech just jumped in and launched their product without completing phase 3. So Valneva's product was completed phase 3 trials in the UK. That was tested in UK, made in UK, but never came out because it was too late at that time. We also did Valneva tested with the mix and match COVID trial when AstraZeneca was in trouble. We also had a clinical trial checking if people who had the first dose with AstraZeneca, how about if we boosted with Valneva, how would the response be like? But that's the actual formulator for that technology behind Bharat Biotech. The product is now approved by MHRA, but it's not bought by the Public Health England. So they also used a dual adjuvant system. And CPG 108 is already been used in Hep B vaccine before. So it's not a new adjuvant, it was already out there, but it's a patented product and they licensed in for their formulation. There was another interesting vaccine came out from Medicago, which used the GSK's adjuvant A03, which GSK used in their pandemic swine flu vaccine about 10 years ago. But that vaccine also did not produce very good clinical efficacy, or if you like, immunogenicity profile early stage, and it didn't came out to the success. Sanofi did a venture with a recombinant protein. So this was a protein vaccine, subunit protein vaccine, which was produced by the genetic recombinant technology. So again, you coat a living organism like E. coli, and then the organism produces, for example, spike protein, and those spike proteins you purify, isolate, formulate, and inject it. So your body is not producing it like mRNA, but you produce it outside. Same technology like mRNA, but your body doesn't produce it, you produce it outside in a petri dish, and that's been injected. Unfortunately, this was also delayed in their discovery pipeline, never came out. So GSK started to work with clever biopharmaceuticals in China to try to push their product out and see if they could work with different companies and out-license to get a product out. Another interesting product was Novavax. Lots of immunologists love this particular product because what they do is they make viral-like particles. Again, it was an insect-based cell line, so they use insect cell line to genetically modify and produce COVID spikes in a flower-like structure. So you have a COVID-like virus, but without the core. So you had 3 spikes popping out from there, a bit like this. And this was adjuvanted by Matrix M. Now the magic ingredient in Novavax is this adjuvant. And guess where this comes from? A natural bark of a tree in Chile, in South America. What that adjuvant does is it potentiates your immune response against an antigen. So it's a very strong stimulant, immunostimulant, and combining this with the antigen increases the efficacy amazingly great. So when Novavax clinical trial happened, it happened through Manchester University hospitals near us, and lots of colleagues participated in it. We were surprised that during clinical trial we had Omicron at that time, and this vaccine was working against Omicron better than any other vaccine at that time, just because of this magic ingredient, Matrix M. This vaccine never came out in time, again, because it was a natural product and it was lots of controversies, cutting the trees in Chile. The government was not supporting, they didn't have enough quantities, and Novavax never had a product in the market before. So they did not have the supply chain and support which, for example, BioNTech had with Pfizer. So vaccine wasn't produced by Pfizer. It was made by Pfizer, but it was designed by BioNTech. So they did not have somebody like Pfizer to run it out in a rush, like in pandemic, where everybody was racing to get the product out. But beautiful product, now approved by MHRA again. Product is out there, ready. Now it's been manufactured, and Serum Institute has managed to get a out license to manufacture in India for that product as well. So they've stopped manufacturing AstraZeneca. Now they're producing other vaccines, and they've been negotiating contracts cleverly with these formulators, trying to get different technologies into their country. So that's the clever thing which industry should do. So you do not take the same product, you take a different product with a different manufacturing technology, and in licensing brings you that technology transfer. And this is one of the things Pakistan should focus on. We don't want to reinvent the wheel. I understand that the discovery cost is very high, but what we could have done is work with these global players who needed support at that time. They couldn't manufacture it, and there was no competition at that time. But we were very slow in reacting and approaching those partners. You all know about the mRNA vaccines, so I'm not going to go in that detail. But one interesting case is CureVac. Have you heard about CureVac? Not many people know about CureVac. They are the mRNA people, basically, right? They, again, German company like BioNTech, and they had an mRNA candidate as well, which didn't quite work in phase 1 very well. They modified it, and they had a very good response. But at that time, they lost the market. So what they did, now very good strategy, they decided to dump it. So nobody knows about that. So they made a product, they went through phase 1, phase 2, phase 3, dumped it. Very clever, thinking, it's too late. Let's move on to next generation COVID vaccine. Now they will be the first one to come up with a bivalent vaccine with GSK. And they have got a combined beta and delta specific mRNAs. It's a dual mRNA system, which is now coming out very soon. So they were a bit slow. Again, you need to understand the niche markets, where the next phase in the market going to be like. So sometimes losing something is better than just keep holding it for a longer period of time. It's more costly. Yeah. So although you have a loss, but having a small loss is better than having a bigger loss. So when I was in Abbott, at that time our CEO was Miles White, and he used to say that our strategy in terms of Miles White is to ditch a molecule as soon as possible in our R&D pipeline. And I used to think like, what he's talking about? Why he want to kick out molecules from the discovery? But I understood later that he meant to say that don't waste time. If it's not working, move on. Invest in something else. Don't just keep holding the same thing. But we scientists do not like to give up, and we keep digging in until we get success. But sometimes that success is not useful because it's too late and nobody is there to benefit from. So strategy, marketing, business development goes in line with drug discovery. Often scientists get upset when a product is striked out, but it's important to work as a team and find the niche areas, because industry would not be funded then, and you need to make sure you are financially sustained, because government not going to fund you like Public Health England did to other companies. So it's a different model in Pakistan. So AstraZeneca case again, CanSino Bio, different adenoviral vector, J&J, and you know the story, what happened to that. But Sputnik, again, very good case study here, that what Russians did, they knew the problem with the adenovector. So they had a dual adenovector system, 85 and 8026. So after first dose, they knew that the body is developing antibody response against the vaccine, and there's no point boosting it, which is why Johnson & Johnson said, all right, we'll go with a single dose. It wasn't a unique vaccine which works with a single dose, because they learned from AstraZeneca. They said, look, booster doesn't work, basically. So let's go with a single dose program. So they changed the dosage, went to the FDA, and went ahead with a single dose vaccine at that time. But what Sputnik did, they used two different vectors. So when you had the first dose, your body learns the vector, produces adeno-specific antibodies. So when the next dose comes in, body kills the vaccine. So they changed the vector, saying booster would be 85 instead of 8026. Now body doesn't know 85, so again you will have a very good immune response. That's why some people would say, well, I have the Russian product and my antibody levels like on the roof. That is why. But 85 and 8026 are commonly found in human population. So roughly say 40-50% people will have pre-existing antibodies against adenoviral vector. So when you have a vaccine based on 85 or 8026, your body might not let the antigen work because it might kill the vector. And this was shown in phase 1 clinical trial from CanSino Bio and AstraZeneca both. And data is publicly available. So the trouble is, sometimes we ignore the science. So science is very important, and better we understand these formulations. In Pakistan, we can make clever moves. We can work with the right partners. We can pick the right products. We can bring them in Pakistan. So when the remdesivir came in, the biggest mistake we did was in Pakistan that we followed the same clinical regimen as we did in UK and US. But we didn't think about why UK is not giving it to everybody, because we couldn't afford it. And it's a publicly funded organization. With the antivirals, you give it within 24 hours, full stop. Otherwise, there's no point giving it. If you were doing it in private, you should have been wise to use it differently. Then you would have seen a magically different response. Similarly, favipiravir was there in Japan, but because UK and US wasn't using it, Pakistan didn't take a notice at that time. We reacted afterwards. There were many brands and products came out, but it was too late, a bit too late. So understanding science of any pathological condition at the point and making the right decisions, good for investment, but also good for public health. And in Pakistan, alhamdulillah, I would say we were very lucky to have 5 different products. In Pakistan, we had the whole inactivated virus vaccine, we had the dual Russian product, we had the Chinese adenovector product, we had AstraZeneca, we had Pfizer, we also had Moderna. In United Kingdom, we did not have a choice. Everybody have to had AstraZeneca in the beginning. And then we were being given Pfizer and then Moderna. So we didn't have that choice, despite we spending millions from the public pocket to produce the vaccine. So Pakistan was still very better off in terms of having that availability and choice. However, people think about these genetic vectors and talk about, oh, it's been developed very quickly and it's a very short span of time. No, the first ADV vector vaccine was from Merck, and they received FDA approval just before pandemic in 2019, and that was for Ebola. It was exactly the same viral vector used for the Ebola vaccine, and it took long time to convince FDA that it's worth approving the vaccine because efficacy was a bit iffy. At that time, we knew in 2019 that antibody level falls in 6 weeks. So this is not something you learn in pandemic, right? So when people were talking about waning immunity, we were like, so what? We knew it's gonna happen. Look at the Ebola data. It's the same formulation. And then Johnson & Johnson got an approval in 2020 in July. Everybody's struggling with COVID-19. In July 2020, they got an approval for Ebola vaccine. Same design as their COVID vaccine. They repackaged it with a new antigen. COVID vaccine is ready. This is the story behind how that quick development happened. So learning from experience and using the platform technologies to develop new products within the industrial capacity. So they did not invest the same R&D expenditure as Ebola. Ebola was heavily, very, very expensive program, and there was a very small return from the market. But that helped them to produce a COVID vaccine, which had a much wider global perspective and then return on investment. So in terms of next generation COVID vaccines, now what we are trying to look at is see what happened with the vaccines, what adverse events we observed, which parts of populations reacted badly to the vaccine, why, and how we could then personalize it. So looking at immunomodulatory epitopes, so for example, the viruses had some epitopes on their structure which causes immunomodulation or dysregulation in the immune system. So you have cytokine release syndrome, you had a cytokine storm, and then this hyperimmune response is basically very devastating for multiple organs to fail. Within the vaccine, because we're using the same antigen, so now we're trying to see, okay, which epitopes we try to delete from the mRNA code to make vaccines more safer, basically. And that's what we are trying to do in terms of discovery. And then we have got the clotting issues, the prothrombotic response from the vector. So is it based on 85, 8026, which is what I think is the main cause? But there are epitopes on the virus which is hypercoagulations as well. For example, if you look at the cause of death for the COVID-19, one of the main cause is hypercoagulation in the lungs. So virus itself is a procoagulatory, and using that antigen will have a knock-on effect. Obviously, you're going to have the same clotting issues. So trying to see how we can minimize it again by using maybe genetically modified antigen, which is not very prothrombotic. So people who have got clotting disorders, who have a stroke before, has coronary artery disease, or had a clot formation in the past, or population are using anticoagulants or the regular therapy. What are we going to do with them? They're going to react with the vaccine. So how we can make it safer? So now pandemic is almost over. We need to look at those niche population groups, the people who are vulnerable in the society, but also they are more likely to react with the vaccine as well, to see if we can personalize different product for them. So in UK we didn't have a luxury before, but now we have multiple products approved, and we might personalize it accordingly. So again, antigenicity versus tolerability is a trade-off. So if you make vaccine more safer, you lose the efficacy. If you try to increase the efficacy, it becomes a bit more toxic. So it's a trade-off between what you can afford and how much toxicity you can live with. So there cannot be anything 100% safe. So lots of my colleagues, particularly clinical colleagues, were trying to cater vaccine hesitancy in South Asian communities, and they would come, oh, vaccine is 100% safe. No, sorry. Even paracetamol is not 100% safe. It causes, you might be surprised that hundreds of deaths every year happens just because of paracetamol overdosing and the liver failures and hepatic issues in children. So such a simple molecule, it's not 100% safe. So it's very wrong to mislead public by saying these kind of statements, because when something happens, they will lose your trust. So we need to be very honest. We need to work on risk versus benefit equation for every patient. So they need to be told, and then they will make their decision. But nothing is 100% safe. All medications have some sort of effects on the body. And then the multivalent vaccines, which I just talked about, for example, the CureVac. There is also a product in the pipeline where we are trying to develop a combined booster dose for COVID plus flu. And that would be very good strategy on public health point of view, because you could have one product every year before the winter season, and that would be more convenient, particularly for elderly population. And then we have the nasal jabs for the kids. So I've mentioned you about the flu vaccine for the children, and a similar COVID vaccine is being produced for children of school-going age, which will be administered through the nose. But it's not an injectable vaccine, it's just a spray, which is a thixotropic spray. And what does it mean is, when you spray it is liquid, as soon as it hits your mucosa, it jellifies, so it becomes a gel, so it doesn't drips off. So we had lots of issues with the current vaccines. It does prevent severe disease, it reduces the risk of hospitalization and death, but there were arguments that it relies mainly on circulatory antibodies, for example IgGs. It doesn't produce IgAs, for example the the antibodies on your mucosal membranes like rangers, protecting the borders. So if you don't have the border security, but you have army within the city, what's going to happen? The enemy will infiltrate the country. You will knock it off at the end, but you will have more damage. But if you put the army at the border, you'll try to diffuse the threat as much as you can at the borders. And if something infiltrates, your IgG would encounter. So that's one of the argument with the injectable vaccine. And some companies were trying to have a combination approach. They were trying to say, okay, you start with a nasal vaccine as the first dose and boost it with an injectable product, and then you might have a broader coverage. And CanSino Bio was one of the companies to run initial trials on a combined system. So we had, the risk of catching the virus in the nose, because you can't prevent that, but you might not fall severely ill, but you will catch the virus. And because the virus is there, it's multiplying in the nasopharynx, you will pass it to the people in your family, in your community. So the community spread is still an issue, particularly in the school age-going children. So we had the flu vaccines and the nasal product, and this is some of the data on flu and Stentra. You will be surprised that these clinical trials, if you look at the strains, the clinical trial, the clicker doesn't work if I go on that side of the stage. Right, so if you look at the clinical trial data, you can see it's the efficacy range. So it's a head-to-head trial versus injectable flu vaccine and nasal flu vaccine in children. And if you look at the data, efficacy is amazingly great for the flu vaccine. So lots of people say, look, flu vaccine never worked, you always get it, so what's the point having a vaccine? It was an issue. So the real world efficacy for flu vaccine was around 40 to 50%. So when Pfizer came in, 95% efficacy, again, I wrote an article in British Medical Journal saying, look, this is ridiculous. This is not going to happen in reality. And I wrote 10 reasons why. And people still look at it saying, well, we are right at that time. So I wrote it in November 2020 when the vaccines were launched. Again, how did I know? Based on previous knowledge, based on the history. So when we moved from IP, injectable flu vaccine, to the nasal flu vaccine, efficacy increased to about 64% in 2019-2018 flu season, just because of a different formulation which provided that community support. And that data is from Public Health England. You can have a look. So now, in terms of COVID, you had Lancaster University completing their clinical trial for nasal jabs, and they've given that to Covaxin in WHO, and that vaccine will be available to all developing countries. Made in UK from a university, and the person who designed this formulation is also a Pakistani-based immunologist working in Lancaster. Very good product. Another one is made by AstraZeneca, because they had a lot of experience in making nasal flu vaccine in UK for the children. CanSino Bio have a product. Sputnik is also making a product. Bharat Biotech has a nasal product. And a French university with the National Public Health Institute is also having a product on nasal flu vaccine. So there are multiple products coming out, and one coming out from Taiwan. And they all are in the late phase 3 clinical trial at the moment. So again, one size dose doesn't fit all, and sometimes we don't have a choice. I do appreciate that. But when we have a choice, we need to rationalize the therapy to make sure we personalize it as much as we can to improve the efficacy and improve the safety on that patient as well. So the other important area I want to talk about is the monoclonal antibodies, which is quite neglected in the past by the pharma industry like India and Pakistan. And these monoclonal antibodies are very important therapeutic response and as a product for many type of conditions, not just COVID-19. They are specifically designed, engineered antibodies. They're monoclonal because they have got only one epitope, and they bind specifically to a target, which could be an antigen, which could be an enzyme, it could be a protein in the body. So you design it molecularly, it locks in, and this is the as much as accurate you can get in terms of targeting the tissue. And the drugs and small molecule therapeutics works generally. So lots of people talk about, oh, I had a tablet for my backache. I've got this tablet for tension headache. So lots of branding in United Kingdom. You can go for Nurofen headache relief, Neurokin backache relief. There's lots of products. And when we talk about this in university, we start talking to the students saying, how does that product work? And in pharmacokinetics, I try to make this joke saying, look, this goes around from your head to your toe. It's not for your neck, it's not targeted. Which is why lots of cancer products have got these side effects, because you cannot specifically target the tumor. You cannot target the cancer tissue, which is why these new cutting edge therapies are coming in. You're trying to focus it on the target tissue so the drug is not binding to other receptors or other tissues in the body, and that's going to produce lots of adverse events anyways. So monoclonal antibodies are very specific, but not 100% again. Sometimes they have a cross reactivity as well, which is why they're very effective, very potent, but sometimes their side effects are very, very severe, because they might have a cross reactivity, they might bind to something else, and here you go, you got a very different response which you didn't anticipate it, along with the therapeutic response. So what happens with this antiviral drugs is very neglected in the world. Historically, antibiotics, antivirals have not been funded for last 30, 40 years, 50 years. And that lack of funding has caused a big issue with the super drugs, super bugs, for example, multi-resistant drugs, multi-resistant bacteria and organisms where the antibiotics are no more effective. Same happens with antiviral drugs. The viruses develop resistance against antivirals, and then the antiviral becomes ineffective. So we have not spent a good amount of money in investing and developing new molecules, which is why the world is at a threat of these variants, which are drug-resistant strains. But you will be surprised that the antivirals have been invested a lot by the pharma companies in the recent years, and I'll show you some data in the next part of the presentation about the opportunities for Pakistan, particularly where Pakistan can invest in terms of new products. So in terms of Pakistan, we have a different situation here. We don't have innovative companies. Some large pharmas we have in Pakistan do not invest in R&D infrastructure within Pakistan. We do not cater for the needs of Pakistan. We just produce products. So how the generic pharmaceutical industry in Pakistan can make some difference in terms of answering public health questions, and that will differentiate you from other companies. So every company has a huge marketing budget, but how you could cleverly do some work in addressing Pakistan's public health questions and then position your products exactly on those domains, and that will give you a competitive marketing edge, so you don't have to compete with 10 different products in the same area. So one of the important areas is access to medicines, which Professor Zahir is very expert about, and he wrote many books on equitable access to vaccines and products and pharmaceutical products. And this is where the industry is already doing a lot, making generic and affordable medications. But looking at the diabetes burden in Pakistan, looking at the cardiovascular diseases in Pakistan, looking at the liver diseases in Pakistan, obviously US, UK not going to do anything about it. It's not their problem. Same issue with the MERS virus. It's not their problem. It's a regional issue. So you can't just sit and wait for something miraculous to come in. What we need to do is work with public health professionals, try to understand the disease dynamics, and a big issue is the infectious diseases. So biggest load in the liver diseases comes from infections. So liver infections, a biggest cause is hepatitis virus, turns into different things. It might manifest into hepatocellular carcinoma in few years timeline. Everything starts potentially with a seed, and often lots of liver diseases are started with a viral infection of some sort. So understanding the epidemiology of diseases in Pakistan, working with national health institutes and understanding the viral variants, the bacterial variants, is your antibiotic is effective? You don't know. How many of you run culture sensitivity on your products? I can bet nobody does on batch to batch. You keep sourcing your antibiotics from different suppliers, you get it from as cheap as possible, you have a QC right, but do you do antimicrobial efficacy? No. Some companies who does it, they do on library ATCC cultures. Have they tried to check on clinical isolates? No. So you're not doing the research which you could do. It's not a very expensive research. A small microbiological laboratory can just do it in some thousands of rupees. Culturing the plates, putting the disc on, see is it working or not. And lots of clinicians, I bet you, will be interested in that collaborative work to see what's happening in their clinic. Why these patients are not responding to your product. Do they need a combination product? But which combination do they need? And lots of good hospitals, clinicians do, and the culture sensitivity test for their patients. But as an industry, we stay behind saying, well, let's not talk about it. It's not going to be helpful at the end because we are not catering for the society. Maybe you want to move from molecule A to B because your cholera maybe in this season is more sensitive to that particular combination. And that's profitable anyways, so you've got nothing to lose. But you reposition your product based on what's happening in the society. So it's a very good case where you can work with clinicians, your partners, you can work with public health societies, organizations like NIH. You can understand how the bacteria is changing in different parts of Pakistan. Maybe some antibiotics are still effective in the north. So you don't want to give them broad spectrum, cephalosporin in that region. But maybe some parts of the interior areas have got a superbug, which might not respond to stage 1 antibiotics. So antibiotic stewardship is important, and a personalized approach is very important, even within Pakistan. And this is where your marketing and sales force can work with clinicians, work with university researchers, and try to develop this data set. Data is evidence. Evidence is power. Once you have that data, once you have that evidence, clinicians are going to listen to you. The regulator can't say no. But data talks. And this is where I think Pakistan pharmaceutical industry should invest a little bit money on some of the marketing budget on that evidence generation side. I'm not asking you to do basic drug discovery research and try to develop a new molecule. Just understanding the disease dynamics in Pakistan and come up with a very clinically relevant approach to treatment. So malaria vaccine, very good example. Malaria, biggest problem in Pakistan. Have you heard about malaria vaccine? I bet not many people think. Never achieved the efficacy targets in the past. Many vaccine candidates failed. This one passed the efficacy target. This one 2021, just last year. And what's the magic ingredient? Again, matrix M. What they did was they used the adjuvant from the Novavax vaccine, the bark from the South America Chilean tree. It's a saponin, and that increased the efficacy amazingly great. So first time malaria vaccine achieved the efficacy target. Not just achieved it, it just went over to the baseline given by WHO. So again, learning from what's happening in the immunology, learning from what's happening in other vaccine areas, and coming up with these innovative ideas. Now, where is the biggest market for malaria? Africa and Asia. Definitely not in Europe. And our industry needs to be taking the initial steps, reaching out to these innovators, out licensing these products. And if you can make it in Pakistan, that's the best thing to do. Invest in now on technologies which can make 10 products later. So your investment is not for one product. Think about these clever ingredients which are now being repurposed and used for hundreds of other products. So think about mRNA technology. How much potential in mRNA technology. It was never meant for vaccines. It was meant for very, very sensitive diseases, rare conditions, cancers, autoimmune disorders, genetic disorders. Thalassemia, for example, we could never cure it. We can cure it now with short interfering RNA technology. We can manipulate the gene, fix the problem. Think about the broad spectrum applications. And taking one technology which is very, very clever could be financially rewarding as well. Even if one product fails, the technology can be repurposed for other products. So as an industry, you should be investing on those areas. Another example is the polio. Now, that has been very controversial. When I talk in public, I don't talk like I'm talking to you. You all are healthcare professionals. So we knew OPV had a problem historically, but we don't talk about it in general because people in Pakistan get scared, they're not going to have a vaccine. Oh, no, 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 I'm not going to have a vaccine. And oral polio vaccine always had this hesitancy in the society. Mothers were not happy to take their children for the OPV. But scientifically, what we did was also wrong. We started putting OPV into everybody's mouth. If a child is fully immunized with an IPV on their childhood immunization chart, they bloody do not need OPV. We don't do it in UK, so why we do it in Pakistan, right? So people are paying out of pocket, as Professor Zaid was saying. Lots of people are spending a lot of money for healthcare, and they're having the, UK equivalent products. So we don't need OPV for them. They are perfectly immunized. But by putting OPV into everybody's mouth, we have caused a wide spectrum exposure, and the vaccine drive strain has gone out in the community and caused problems historically. But because in Pakistan we had higher number of wild polio cases, so the risk versus benefit again was favoring OPV, right? So we always preferred it because we couldn't immunize remote areas, rural areas, parts of the country. But what's the point putting OPV in urban districts where people are fully immunized? It's live attenuated vaccine, contraindicated in immunocompromised children, but a campaigner would come and knock on the door, put it into everybody's mouth without checking if the child is on steroids, if it's immunocompromised, the child might have got kidney transplant. You can't put live attenuated into somebody who do not have a good immune system. And that is the problem in Pakistan. We do not understand things scientifically. We run into trouble. Now, there is a big push to get rid of OPV, and change the global campaign to IPV. Why? We had a trouble in UK. And we don't use OPV. But what happened? The immigrant community who had OPVs overseas come into UK, live in London, and they spread the vaccine-transmitted virus. We never had a case, and yet we had a trouble in this year. There's a big push from UK, get rid of OPV from every part of the world and go to the IPV. In the past, we would say, oh, who would going to use injections, cold chain problems, supply chain issues, who's going to put it in a cold chain? How are you going to guarantee cold chain? Now look what happened with COVID. You delivered COVID in every remote district in Pakistan. So when there was a need, you could do it. You did it. Now infrastructure is present now. You got to use it now. Otherwise, in public sector, it will die. So this is the time we get rid of OPV, go to the IPV, and it's a better vaccine. It has a more efficacy, and it will help Pakistan eradicate polio forever, like we did in UK. So this is the area where Pakistani industry. Now I heard in June this year, some, I'm not name it, but some investors in Pakistan approached us for a consultancy on OPV manufacturing in Pakistan. And they were like, oh, we're going to manufacture polio vaccine in Pakistan. First time, homemade vaccine, da 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 da. And I was like, OPV, forget it. It's being phased out. You don't want to spend money on it. And by the time you will have a product ready, it will be discontinued automatically. So again, looking at what's happening in global stage, understanding the viral dynamics, and trying to come up what's good for Pakistan. And this is what Pakistani industry should do. Obviously, it should have a business case, because industry would not survive if it's not financially viable. So no argument about it. But within that spectrum, we could cater for our healthcare needs. And this is the true role of the pharma industry in society, to provide for the society, to provide for the healthcare needs of the society. So this is a very interesting slide showing you an evidence that how antiviral products are, exponentially growing. So if you look at the timeline here, 1960s, we first time has human papillomavirus, and then we had the first antiviral being discovered to cater that. And then we have HIV, then we have HCV, and as the viruses come in, you can see the number of antivirals increasing exponentially. So in the past, we might have only got one drug, and in 2016, we had 41 antivirals in pipeline. Now this is until 2016. What happened during pandemic? Amazing investment. You might remember aciclovir. I know this because Abbott used to make it as well. That was the baseline. You started with that one. Again, another Abbott example, Calitra, the combination pill. And I was very lucky back in, when it was, 2013, 14 probably, I went to Ludwigshafen in Germany and I saw how Calitra was made for the first time. And nobody could make it because it was a Maltrex technology. It was not a simple tablet. This combination antiviral pill was manufactured using a very special hot melt extrusion process where you recongeal this liquidified solid material. And that was cleverly done to improve bioavailability, absorption, because the antiviral has the biggest problem. Its pharmacokinetics are like this. So sometimes you have this tiny blood concentration, sometimes you have this huge concentration. So intra-subject variability and then inter-subject variability. So person to person and within the same person. It never works. And that formulation resolved that issue. So that a clever combination, one was a CYP3A4 inhibitor, so improving the efficacy of the other, and that was a gold standard afterwards for HIV. So learning from that, you see this combination, very clever. Pfizer used same technology for Paxlovid. So they combined their nirmatrelvir with the same ritonavir combination to improve the efficacy. Anybody remembers the aciclovir dose? How much do you take per day? More than 1,000, right? Anybody remembers favipiravir dose? 1,400, 1,500 milligram. You can't have, enough absorption and circulation. The pharmacokinetics are too poor. But look at Calitra, what happens? You work with a clever combination, you work with a drug interaction which is in your favor, hence the Paxlovid dose was very small compared to molnupiravir, very high dose from the Merck. So you had then remdesivir intravenous available during pandemic, and again it wasn't very well placed for COVID patient because we couldn't fund it in UK. It's a public health funded organization. We couldn't give it to everybody, so we reserved it for hospitalized patients who might not recover, and we said, all right, we'll just give it to them. But we couldn't give it to people within 24 hours. But as soon as Paxlovid came, you can think about the dosage regimen completely changed. But this was not the first time. Think about the product for the flu. That's been given in the same regimen within 24 hours. So remdesivir, unfortunately, at that time wasn't used appropriately. Avigan from Japan was neglected by many countries as well and was not used, although Japan government offered royalty-free production for favipiravir in 2020 to anybody who wants it, but nobody benefited because maybe Western countries were not using it. They had their products in the pipeline, so they had different focus. But countries like Pakistan, we can think out of the box, understand the disease, and come up with strategies which will work for us. And products were there. We just did not use it. Molnupiravir is a very beautiful example. This drug was developed for a type of bloody influenza virus, a hemorrhoidal influenza, which mortality is very high in elderly patients. This molecule was developed in a very small university in US, and nobody wanted it at that time. Nobody wanted it. So a small biopharmaceutical company did some trials in ferrets, and they found that the drug is very good, effective. They took it for peanuts, and during pandemic they sold it out to Merck. They made billions. And when they did trial on molnupiravir at that time, they found that it stops the replication within 24 hours in ferrets. So you give it, and then within 24 hours virus will not multiply, which means next 24 hours if you put healthy animals around, they will not get infected. But we did not invest on that molecule, gone to Merck at that time. Now it's difficult to negotiate with a big company, But previously in 2020, I presented molnupiravir. I remember in June 2020 for the first time saying, look, invest on that molecule. It's available. The data is very promising. All the phase 1 trials were done, and that was a safer investment. Same happened in favipiravir. I think it was the PPMA webinar online, if you remember, in November 2020. It was again PPMA platform. And then the Paxlovid, very difficult product, very nasty product, 150 drug interactions because of the CYP3A4 mechanism. But you can think of how financially viable that product was and how clinically important this was to protect somebody who you know will struggle with COVID. So you give it on day one, don't wait. You know that patient has got maybe pre-existing conditions or a high age or already had kidney fail, can't afford to have a viral infection like COVID. Definitely within 3 days patient going to go in hospital. You give them in within 24 hours, no chance taken. So some future perspective. Shall we take a break for a coffee, or shall I continue, or what would you prefer? 1 minute break. We'll take it. So any questions for 1 minute rather than I start this part of the talk? Any questions? I can take a question in a minute.
But unfortunately, none of company could get the registration on time. And now, as we all know, that 7 different variants have evolved, and there is an apprehension that which variant will attack which person, and probably that particular therapeutic modality will work against or not. So those molecules are strict. In the medical fraternity, there is a consideration that which molecule will be really effective out of those 7 various mutant, corona variants.
That's a very, very important point. I'm not trying to undermine any population fraction in the society or prejudice against somebody, but historically we know that immunology tells us that the variants are more likely to be produced in somebody who is immunologically weak. So if someone has a weak immune system, very elderly or immunocompromised, they are the largest production factory of variants, and those fraction of populations were neglected in COVID-19, which is why we had that trouble. So even in UK, we did not had at that time Paxlovid, we did not use favipiravir, we did not invest on antivirals, and COVID task force was so heavily focused on vaccines. They thought vaccines will magically cure everything. But we also needed multiple strategy. So UK realized it very quickly, and then they started investing on antivirals. They funded it heavily. They funded monoclonals. They funded vaccines. So they did it right. So UK was a very good example of a public health organization who invested from public pockets into 3 different dimensions to make sure that you cater for every angle. But we didn't do it in time. So if we go for a personalized approach, and we try to protect those vulnerables in the society, you're not just protecting them, you're also protecting society, because variance comes from there. Your second point about regulator is also painful. I understand that, but what happens, say, for a regulator, if you come up with a proposal, you have a conflict of interest. So if you bring a proposal, I know you're going to make money out of it, so how would I trust you? And that causes problem. So we need to work together. You can involve people like clinicians, you can get involved like academics like us, and we can sit with the draft, we can sit with those organizations and try to educate them in terms of what's best for the Pakistan, what's best for the society. And they also need to understand the risks associated with it, because at the end of the day, DREP would not like to take responsibility of something novel coming out from Pakistan which is not going to be safe, and then the whole world are going to look at Pakistan saying, look at that, what they have done, and they're not going to do that. So how we would mitigate the risk? I'm sure industry would have done a lot of hard work before bringing a proposal, but there is a conflict of interest. There's a genuine conflict of interest. So we need neutral people on regulator. So regulator alone can't make that decision. So for example, in UK, we have JCVI. JCVI 90% is independent clinicians or academic scientists, and they advise, they make recommendation. Then government has an authority to either listen to them or not to listen to them. Legally they are not bound to listen to them, but politically if they don't listen to them, it's gonna be a massacre for them. So they will listen to them, they will always listen to them. So although legally they have no authority, but because they have an independent scientific foundation, the regulator have to listen to them because it's their scientific opinion. And that opinion, as Professor Zahir said, is not rigid. It changes with time. JCVI changed their vaccine policies even twice a year based on new data set. So they were not ashamed or embarrassed saying, oh, we said it this 2 months ago. How are we going to look in public if we change our statement? No, they were very open about it. Said, look, now new data suggests this, let's do this now. So I know regulators are hard, but we are happy to help. If you want to have that dialogue with the regulators or develop those independent bodies, because pharma companies could fund those initiatives where the clinicians and scientists can come together, make those bodies which governments can then listen at the end. Again, we need to remove the bias and conflict of interest. We need to make sure that we are not paying for the lip service, which is what regulator thinks. It's not always true. I know lots of companies do a very good job and they're doing a great work with clinicians, but this is the perception, and we have to change the perception and also cater the bad practice in some parts of the industry. So shall we break for coffee now? Yeah, it should be all right. And then I've got last part of the talk. In this part, I will present some ideas within the generic sector as to how Pakistani industry could benefit from innovations at a low cost, for example. Right, so in this section, I'm going to talk about some of the opportunities for Pakistani pharmaceutical industry as a country, as a nation, what we can do, where we can invest in, and what would be the cutting edge therapeutics for tomorrow. So some of the ideas I've already rolled out in the previous part of the talk. So one of the areas where now the drug discovery is going in, is everybody is aware of, is the biologicals. And Pakistan traditionally has been more focused on generic products of small molecules, but the world is now moving away from small molecules and going to the biological products. I know biological products are difficult to make. The infrastructure is not present in Pakistan, but I think it is the right time that we invest on those products and technologies rather than competing for low margin, highly competitive generic markets, even within Pakistan. So the pharma companies, now is the time to think, and those who will diverse now, those who will invest now would survive for a long period of time. Otherwise now, it will be very difficult to keep surviving and competing in a very highly crowded small molecule generics industry. So biologicals could be peptides, it could be proteins, it could be hormones, it could be any biological product which is not just a small chemical drug or entity. Because biologicals are not just a small chemical compounds, their efficacy and their quality control cannot be tested by just simple analytical techniques which we normally have in Pakistani pharmaceutical companies, which makes one of the biggest hurdle in bringing those products in, because we can't just do a UV spectrophotometry, for example, or HPLC assay and say, all right, the product potency is good. It's a complex molecule, and often potency is lost just because this 3D conformation is changed. So if the molecular structure is same, molecular weight is same, HPLC assay is perfect, but this protein chain is slightly tilted, it will lose this potency completely. It will not bind to the receptor. So it's a very different board game, and you need a step change in investment. You cannot just change little bit and start making those products. But this investment worth it because this can go longer and it can go wide spectrum. Once you have the right technology, you can make multiple products. And because many countries do not have this capability, if you go back say 50 years, Pakistan used to export their pharmaceutical products to many countries. Competition has increased, now competing in those markets are hard. And lots of companies like UAE, Egypt are now making generic copies themselves. But nobody is doing biologicals. So if Pakistan leap into biologicals now, we will gonna get our export margins again, because other countries not gonna go very quickly. So Pakistan was leading lots of low middle income countries, say 20, 30 years ago, 40, 50 years ago, and we were the largest producers of medicines outside the mainstream Europe and America. But with the competition now our stake is very minimum. So we can regain that share by going into the niche products which Egypt can't make it, which UAE can't make it, which African countries can't make it. We could now leap into that. Vaccines is another area People argued in the past that vaccines are produced cheaper in some parts of the world, for example, India and China. So what's the point Pakistan makes a vaccine which is expensive than internationally available vaccines? So financially they were never viable. I accepted that argument, but that was the past. The game is now changed, and innovative solutions for the modern challenges means that investment in vaccines could give you the right product share in terms of international markets. You can regain your export potential, because once you have the technology, for example, say if you invested in inactivated virus technology, it's not just COVID vaccine you are making, right? You could diversify your product portfolio accordingly. And again, your business development and the strategy is the key. So you need to have a very good product development team, the market strategy, who's keeping an eye on the global scenario and investing at the right time to produce a product which is based on the same portfolio technology. Similarly, if you were to decide to go on Novavax technology, for example, which is the recombinant protein technology, think about the Novavax is formulated by the method which can churn out any proteins on a petri dish, for example, or a big tank. So if you can have that technology, you can produce any therapeutic peptide and protein in the same manufacturing facility. You just need a tech transfer. So investing in the right technology area could give you access to 10 different products, not that vaccine. So technically you invested in vaccine, but actually not. You invested in that technology which is making a vaccine. The same technology would have been used in a cancer product, which is coming out. People can't afford it. One dose might cost thousands of dollars. Pakistan can make it. Why it has to be India? Pakistan can make it. It's just because we do not have those technologies, and loss of genetics scares people. Immunology scares people. I don't understand these antibodies business. That's why people don't invest on it. We've never been educated in genetics and immunology much. We had a poor pharmaceutical education structure. Our main focus had always been in pharmaceutics and pharmacology, and we stopped there with chemistry. We don't go beyond. But revamping our knowledge in immunology, in genetics, in small molecules, proteomics is what we need from university side as well, because industry can't do it alone. We need to produce the right graduates. But we have biological sciences degrees, we have biotechnology degree graduates, we have got other areas to benefit from. Genetics is being taught separately, not in pharmacy, but it's being taught separately. And those people can chip in, become part of your team. So you still need a pharmacist, but you need somebody else who knows that area, and then you make a good team. But if a pharmacist knows that, it's a lot better, because then he can connect the dots. Because he knows immunology, so he can formulate an epitope in a formulation using that knowledge. Because he would know the pharmacokinetic as well, and then he can better talk to the clinician as well. So pharmacy is a unique, blend of knowledge which comes from different sciences and makes it into a package which you need as an industry. But if you don't have it, still you can make a team by using different graduates and make that multifunctional area. So lots of people talk about mRNA being now it's become a fashion. But mRNA is not just the RNA therapeutics. Has anybody heard about siRNA in the hall? Nobody. Now, you will be surprised that the siRNA therapeutics came first, even before mRNA. So RNA is not just messenger RNA. The siRNA is a short interfering RNA, which has been used for gene silencing. And in people with rare diseases where there is a faulty gene, there is a defect in the body which is causing a disease, you can silence that gene by short interfering RNA. It's a type of RNA, but what it does, it goes and silences that particular part of the genetic sequence in your body. That means you actually cure the disease. Think about conditions where people can't live a quality life because of that genetic disorder. This can be an answer to that. mRNA therapeutics is only where you want to produce a particular protein in your body. So when you give mRNA, it translates into a protein. So you provide a code, and that code translates into a protein. Sometimes you don't want to produce anything, you want to shut down something. So RNA therapeutics works in a very multiple dimensions. It can work for cancer, it can work for rare genetic disease, it can work for immunomodulatory disorders, hyperimmune disorders, and it can work as a vaccine as well. But it was never meant for a vaccine, if you like. Okay? So vaccine is just one application, but there are many other applications. And this is just a cartoon explaining how synthetic version of siRNA goes into the body. It binds with the genome and silences a particular part of the genome. And one of the products, which some of you might have actually heard, Lacvivo, is an siRNA product. And that was launched a few years ago, even before pandemic. And what that product does is a inhibitor for one of the PCSK19, I think, receptor in the liver, which causes destruction of lipid uptakes. So what happens is that the receptor stops body to retake cholesterol or LDL, for example, from blood to the liver. So you have a very high cholesterol in the circulation. And what this particular product does, it silences that gene, so your receptor is not destroyed, and all circulatory LDL gets stored in the liver. Just after one injection, you have your lipid profile. You say, what happened? Everything is gone, just disappeared. Uncomparable efficacy to any hyperlipidemic drug. Game changer completely. In United Kingdom, this was not publicly funded before, but now the NICE, which is the National Institute for Care and Excellence, has produced a guidelines, and they have now recommended it for people who are not responsive to anything, dietary changes or Lipitor or any statins not working. They have a high cardiovascular risk. Now they have allowed the use of this particular product. In the past, they didn't allow it. It's not just because it wasn't good, just because it was so expensive, they couldn't use it. So pharmacoeconomics again, like Professor Zahir was explaining in the previous part of the lecture. So when you try to read the science, when you try to see what UK is doing, don't just take it as a granted. Try to understand why they've made those decisions. What's behind those decisions? What's the cost-benefit, analysis they have done? And because we are using it as an out-of-pocket expenditure anyways, so our decision-makings are different in Pakistani society. So some products, if not being funded or used in UK, doesn't mean they're not good enough. For example, I give you a very easy case. So atorvastatin, Lipitor. So in United Kingdom, everybody used simvastatin until last 6 years. And this was the first line drug funded on NHS. Everybody would be on simvastatin. Now, simvastatin is a very lipophilic molecule. It has a different pharmacokinetic. It goes into your tertiary body systems, and it penetrates into the muscles and never comes out. And it shuts down HMG coenzyme in the muscles, causes rhabdomyolysis, which is the damage in the muscles. So all hyperlipidemic people with high cholesterol, are elderly people, right? And what we are having at the end, damage in the skeletal muscles. So everybody have got issues. When the atorvastatin was synthesized by Pfizer, their idea was to change the pharmacokinetics. So what they did, they used a dimer to combine two molecules together in a way that it becomes more hydrophilic, if you like. So it doesn't penetrate into the adipose and the muscles. So rhabdomyolysis is very, very minimum. It's a lot more safer for elderly people. And guess what? We never gave it in UK because it was very expensive. We couldn't afford it. But in 2000, I think around 10 or 11, when Lipitor went off patent, it became the first drug of choice for hyperlipidemia in UK automatically, because now it was 20p for 30 days supply as a generic copy. So we could fund it, we could afford it, we changed our regulations completely. But look at the other countries. They followed UK saying, oh no, simvastatin is the first choice. And everybody blindly used simvastatin. And how many elderly people had a poor quality of life just because they did not use the right molecule which was available for donkeys' years. And we could afford it. Lipitor wasn't too expensive in Pakistan. It was very expensive in UK because Pfizer won't sell it cheap to NHS. But it was cheaper in Pakistan. So this is an example of a drug which is a cholesterol drug made using siRNA technology and is present for a few years, even before mRNA product came in. So this is not new. This discovery is happening for 20 years until you get a product. Once you get one product, one siRNA product, you can have a range of products because now regulators are convinced for the safety and efficacy. So the platform works. Once you have proven the platform, when I was in University College London, one of my colleagues was working on similar technology for the lung diseases, very, very rare genetic diseases of the lungs where people just can't live after a certain age. So what they wanted to do is make an inhaler with siRNA nanoparticle. You inhale in the lungs, silence the genes, and your lungs are healthy, revert back to normal function. And that's about 11 years ago I'm talking. And they made it at that time. I don't know what happened. It might have been out licensed to a pharma company, but that's been in the pipeline for a long time. Monoclonal antibodies. Again, the first drug you heard about monoclonal, probably generally speaking, majority of you, was tocilizumab. And before that, nobody knew about MABs. What are MABs? Nobody heard about MABs. But MABs existed for, again, at least 20 years. This was the first one came out during pandemic time. It was interleukin-6 antibody, which was meant to target the hypercytokine syndrome or cytokine release syndrome and try to protect the body organs from immune destruction. Some confounding data in terms of clinical efficacy because you need to find out what's killing the patient. So if you're not giving this product to the right patient, you might not get the clinical benefit. So there's a time window where you can use it, plus you have to assess if this is going to be beneficial to the patient or not. But this was the first MAB, but this was not targeting SARS-CoV, it was targeting interleukin-6, and the drug was produced for other purposes where interleukin-6 was causing hyperactivity in the disease. So we repurposed this for COVID. Brilliant response. The first COVID and cocktail antibody was from Roche, which actually targeted the epitopes on the virus. So these antibodies were synthetic monoclonal antibodies. Again, monoclonal means they have one epitope to bind, but they had two different antibodies in there. So it's like a combination product. So you bind the virus at two different sites. So even one mutates, an antibody doesn't attach, latch on, at least the other one will latch. And that combination was the first one. You might remember when President Trump had the COVID, he came in with a helicopter with that Bollywood scene. He was given this cocktail. This was at that time on clinical trials. It wasn't publicly available, but he received that cocktail of antibodies which neutralizes the virus. So this is like your own antibodies, but your own antibodies are polyclonal in nature, which are formed as a response of antigen or a real virus or a vaccine. These are synthetic antibodies which you make in an animal model and they latch onto their target, which means they can neutralize the virus immediately. So for example, immunocompromised people who are vaccinated but they're not responding to a vaccine, they're still vulnerable. That is a solution you can provide them. So again, your clinical guidance should not be a copy paste from Western world. You need to understand what therapeutics are there, what is the need in Pakistan, do you have a fraction of population who can afford it or who needs it, and you just position that product there and it will work. So another one came from GSK. Again, it was another antibody. To identify the nomenclature, they will always end with a MAB. So monoclonal antibodies, all the generic names of MABs will end with a MAB. So adalimumab was another example I give because it was from Abbott, Humira. Miss Anjum would remember it. And that was, again, ages ago. It was then spin out into hospital business afterwards. But we were making it at that time. And that was at least 20-30 years ago when Abbott launched that antibody, and it still works. Herceptin is another one which is used for breast cancer, and there are plenty of them. And then there was another one came from AstraZeneca. This was also a combination of two antibodies to latch on two different sites of the virus to have that kind of dual safe mechanism. Another technology which probably you would have never heard is CAR-T therapeutics. Anybody heard about CAR-T? So CAR-T is a very amazing, system. So in COVID-19, everybody now know about T cells, right? Because you heard about T cell response, and T cells kills the virus, da, 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 da. So they're part of very good immune response, cellular immunity. So what happens in cancer is your body's immune system or immune cells cannot identify a poorly functioning body's tissue or a cell. So, for example, abnormal tissue or a cancer cell. And if the immune system doesn't recognize it, it keeps growing, you get a malignant tumor, basically. So I have got probably plenty of abnormal cells in my body, but I don't have a cancer because my immune system recognizes it, destroys it. So everybody have that. But when your immune system stops differentiating between good and bad, that's where the abnormal tissue starts growing. So what we do in CAR-T is we take your body's blood sample out, we go for leukapheresis, which basically means you separate the white blood cells, leukocytes. So your T cells are obtained, so your own T cells are obtained. Then I go in the lab, genetically modify a T cell and train them to recognize the cancer in your own body, right? So that recognition which T cells have lost, we train them in the petri dish. After they are trained, we culture them. So for example, multiply them. Then we purify them, sterilize them, re-inject it in your body. And then the T cells goes around and kills the cancer tissue. Very targeted, your own cells, so no cross-reactivity, no interaction, no animal product involved. And you might be thinking, oh, this is just a hypothesis. No, look at the products. The first one was from Novartis, came out in 2017. The difficulty is you have to make it for every patient. So oncologists, will work with you, will give you the sample. The sample goes in the lab, we manufacture it, we make a large volume parenteral, it goes back to the oncology unit, they use it with chemotherapeutics unit, inject into the patient. So tailor-made for every patient, very expensive, but it works. It's right on the target. Again, acquiring that technology is not difficult. The science behind CAR-T is published. That knowledge sits in the immunology. You just have to learn the knowledge and make it. There might be some IP issues around it, which you might have to work around in licensing, for example, but it's not something you can't do. And we can work with hospitals, oncology units. The hospitals, for example, we have in Karachi, a couple of units, Kiran, for example, around the corner. We have Nouri in Islamabad. We have NIH. We have Imran Khan, Shaukat Khanum. We can work those oncology units and do that within the hospital pharmacy settings with the genetics and immunology sector involved. You have the better context with the clinicians. Who will have that? The pharma industry is the best to work with clinicians. You always work with them, and we can have that. So you don't have to have a manufacturing unit, for example, established in the industry. It could sit in the hospital pharmacy. They can do it with you, and you can provide it for the patient. So these are very niche opportunities, and you can see the Janssen's products came in 2023, and they've got different receptors. Most of them are targeting CD19 because they're killer T cells. And what killer T cells do is they destroy the cellular machinery, and that's why you pick up the killer T cells, because they will then eat the cancer tissues. But there, if you look at the indications, there's a range of indications now they've been produced for. So mRNA is not the only solution. There is a variety of technologies and products out there. There's a bit hype about mRNA. It's a future for everything, but there is siRNA, there's CAR-T, there's lots of other products you could diversify yourself on. And always pick on things which people are not talking about, because what's very advertised in media, all countries are bidding for it, but there are secret gems around which nobody thinks about. For example, favipiravir, nobody thought about it at the time. I kept shouting molnupiravir, I kept shouting, nobody thought about it, and then when everybody knew because of the press, it was too late. So there's a risk involved, obviously, because you're intervening early, but you can scientifically calculate the risk. You can see, okay, have you got the animal data? Have you got the toxicology data? Have you got some basic efficacy? Yes. Then it's a calculated risk. It's not a new molecule out of the sky which has never been tested kind of thing. And then the another sector which I would like Pakistani pharma companies to focus on is antivirals. Think about how many companies make antivirals. Abbott is struggling for aciclovir. We have a lyophilizer which is very expensive, very costly. This was costly 20 years ago when I was there. This is still costly there. They might shut it down. But think about people in Pakistan might not have aciclovir at all. What you gonna do then? Can't afford an imported aciclovir. It's very expensive. So Pakistani companies should come forward. And Abbott always struggled because it has a more hierarchical multinational structure. They couldn't make it cost effective. They couldn't do the economy of scale. They couldn't use their machinery to make other things. But you could do it. Once you acquire a technology, you could make multiple antivirals using the same technology. So, for examples, the products I've mentioned earlier in terms of COVID-19, there's a lot of examples which you could use. For example, you have Paxlovid now, molnupiravir, favipiravir. There's a range. There's a Calitra combination litonavir product. It's off patent now, so it doesn't owned by anybody. Once you invest in the seed molecule, all antivirals are slightly related anyways. So investing in the active pharmaceutical manufacturing in antiviral side is not going to die, because you're going to have hepatitis, you're going to have shingles, you're going to have chickenpox, you're going to have influenza, you're going to have coronavirus, you will have respiratory syncytial virus killing babies. You're giving antibodies because you have a multiple infection with bacteria, but it starts with a virus. And it's an issue in pediatrics in UK as well. It's the largest killer for children, the RSC. And you guys can work with pediatricians and bring those products in, which will be more effective than just broad spectrum antibiotics, because it starts with a viral infection and body becomes vulnerable. You get a streptococcus involved, you get other bacteria involved. Now it makes a very complex infection. Kids struggle surviving. So again, talk about high value active pharmaceutical ingredients. So API manufacturing is costly. You can't beat India and China on the cost, so many companies don't want to invest on it. But if you pick on high value APIs, then India, China can't supply the world. So it's again picking the right molecules. If you pick the high value products, you will have a market share. You will have export share as well. You don't have to formulate the product, you can sell the API to the large pharma companies. Because there are some problems working with China and India. And sitting in the UK, I can see those issues. So Pakistan could be an alternative for people who are struggling working with China and India. And we need to encash that opportunity before somebody else jumps in. So last month I was in a meeting at Oxford with some Nigerian FDA, and there was the Director General of FDA talking about bioequivalence center, and she was trying to bring in some cutting edge production technologies in Nigeria. So they're very rich, they've got the money and they've got the drive, and those countries are now coming up on international stage, and they've asked us for some help in setting up their bioequivalence center and developing some innovative products in their national manufacturing spectrum. So Pakistan is leaving behind. And my heart cries out, when I see some countries going so fast. And I originated from Pakistan, started from floor in Pakistani manufacturing sector, seeing the reality. I know the difficulties. So I'm not talking from UK, who has never worked in Pakistani pharma industry. I was working on the shop floor, production floor, QC labs, so I know your problems. But still I feel we can move forward. The difficulties can be addressed. So biopolymers is another area we've never invested in. So Pakistan is very rich agricultural country. We produce lots of agricultural products, but we never make excipients. Why? Because we think, starch is so cheap. It's cost 1p for a kilogram from China. So I'm going to make a lactose and starch. Don't make it. Make a functionalized lactose, which you buy from Europe for $500 a kilo. You can make it in Pakistan. It's a specialized granulated starch, which you can't have it from China. It has a very specific properties, used in inhalation products, but you can't have it from cheaper countries. So you need to invest on excipients, but don't make cheaper products. make high value ingredients. Now think about banana gum, think about mangoes, think about lots of vegetables and products you make. They've got pectins, they've got starches, they've got lactoses, and they've got other carbohydrates which are then functionalized for pharmaceutical applications, and they sell very expensive. They're not cheap. They're very expensive, very high profit margins. And Pakistan's agricultural source produces a big waste on the peels of those fruit products. And you could use that to produce excipients, but not cheap excipients, functionalized excipients which are used in modified release dosage forms, and they have a function in the product. They're not just excipient or fillers. And that's one of the areas you can work on. Another one is a capsule formulation. So I heard there is a company somewhere in Punjab and they make capsule shells, but their market is very small because they're slightly expensive and people buy cheap capsules from China. So how they can survive? Obviously, they can't make it cheaper because they have to be financially viable as well. But what they could do is extend their product to the next levels. For example, can they make an innovative capsules? Can they redesign a capsule shell? So I'll give you an example afterwards, what we have done in Huddersfield recently. So novel formulation platforms and high value APIs, for example. So this is one of the case study I give for your understanding only, but this is not just one product. So majority of drugs are either weak acids or weak bases or they're salts. One of the example I give is ibuprofen. So ibuprofen, again, close to my heart because Abbott used to make Brufen and then it was sold off to Racket Bankiza afterwards, like Strepsils went away. But ibuprofen, interesting molecule, right? Abbott used to make a biconvex sugar-coated tablet. Everybody copied it. So you have 100 generic copies, all same. No innovations, right? And in Abbott, we knew it was a nasty product. It cost money, sugar coating was hard, quality control was difficult. Biopharmaceutically, it disintegrates late. The lag time is high, Tmax is late. The analgesic effect, people have to wait 35 minutes to 2 hours to see the analgesia effect. Rubbish product. But because the pink ibuprofen, Brufen sells, everybody makes it. But look at what Racket Bankiza did. So we used to work with Racket in UCL when I was at London. And what Racket was trying to do is, they were trying to make, all right, look now, ibuprofen is 10p a pack, 20p a pack in supermarket. Can we have a brand which still sells? Because Nurofen stopped selling. Because there were ibuprofen, 10 times cheaper price available in UK over the counter. So they come up for like, okay, we need faster. Can we reformulate it? So we can say, look, faster pain relief. That's the tagline. It's an OTC product. So again, you can bypass clinicians and have a direct consumer-based marketing. And what did they had was a liquid fill capsule, right? So they said, all right, in this one, ibuprofen is pre-dissolved in a liquid formulation. It's in a soft gel capsules, so it will work faster. It didn't work faster. If you look at pharmacokinetics, it didn't work faster because it was a solution, but as soon as it comes to the stomach, it precipitates. It has to redissolve afterwards. So there was no gain. If you look at the pharmacokinetics, but people loved it. Red colored, very bright looking product, and it started selling for a bigger price. Again, Nurofen brand, so people loved the reputation and they made huge money out of it. But with time, the patient become educated, the pharmacist, the doctor saying, well, it's the same, you can still buy the cheaper one, So Racket came back and saying, what can we have now as a pipeline? So they changed it to sodium ibuprofen, which was a sodium salt of ibuprofen. You might have heard the same story with diclofenac sodium, diclofenac potassium, diclofenac diethylammonium salt for the Voltral Imul gel, which you had. Fantastic formulation. Same API, different salt form, emulsifies, absorbs through the skin, higher bioavailability, better pain relief. Same drug. Old dogs, you teach the new tricks. You don't have to rediscover a drug. You can work around clever formulations to get the market in your hand. So sodium salt of ibuprofen is less acidic, so it has a different ionization threshold, dissolves better in stomach, absorbs faster, and it works faster. So there was an advert in London saying you got a headache at Heathrow, get a relief by Hammersmith. So they worked out it takes 15 minutes to get from Heathrow to Hammersmith. So there was an advert on the tube. So people coming on from flights, tired, headache, saying, Get it Nurofen Express, and by the time you get home, you will be fine, kind of thing. It sell. And then they brought another product called lysinate. Now this is 342 milligram ibuprofen lysine. Guess what? It contains same 200 milligram ibuprofen. No change in dose. Lots of people think, oh, it's 342, higher dose. No, no. The rest of the weight comes, 142 is from lysinate. Lysine is an amino acid, it's a bigger molecule, it carries a weight. Equivalent to 200 milligram ibuprofen. Potency is equal to 400 ibuprofen. Highly potent, just because it dissolves better and it does not produce that acidity. Now, guess what? It's not patented. It's a generic API. Who makes it in Pakistan? Nobody, because everybody wants to make Brufen. So thinking out of box, again, educating patients and bringing new products. And the advantage of these products are they're small products, simple tablets. You can coat it, you can leave it uncoated, cheaper to make, don't have to sugarcoat it. Easy, better pharmaceutics, better bioavailability, more clinical efficacy. Remember that these products are only used for acute conditions. It's not used for chronic conditions, because this advantage is only short-term gain. So if I'm using normal ibuprofen every day for 2 weeks or 4 weeks, it doesn't matter. At the end, I go to steady state kinetics, so it doesn't matter. It's the first effect, right? And lots of time people use it for a short period of time. Headache, pain, something happening, cold, fever, flu. They want a quick effect. They don't want to miss their job or university or whatever. So this is the OTC market. And this is just one example. There are plenty of examples like this in UK and US markets, which Pakistan is not in cashing on. And we're still competing on same generics with multinationals. So Abbott never did it. I told them many years ago when I was in Abbott, because Abbott is a big multinational company, they can't change bloody Brufen. But why not you guys do it? You benefit from it, right? So this is another example. So I'm just going to quickly pass through this because I'm not going to go into scientific details. These are the polymers used for enteric coating. So those of you who are aware of the production cycles, you use different polymers to make gastro-resistant tablets, for example, or capsules like Voltral or diclofenac products. Apival, for example, valproic acid is also enteric coating. And you have 10 different polymers, all good, different brands. Some from Japanese, some from German, Evonik. All have different pH thresholds, different types of coatings, dissolve at different locations in the GI tract, and you can target your drug at different parts of the duct. For example, ulcerative colitis, you have UC, inflammatory bowel disease, Crohn's. You need to bring in innovations, where the drug is going. If it's not going to the target, patient not going to get the benefit. And these are the polymers. Normally when you do QC testing, dissolution testing, all polymers will give you the same profile, interchangeable profile. If you look at the clinical trials, they are not interchangeable. They're very different. So what we did in London at that time when I was doing my research, I developed a in vitro dissolution system which could simulate what would happen in vivo in the clinical trial. So you could screen your formulations and pick the right candidate for clinical trial. And that was one of my major projects during the PhD as well. And we did lots of work for pharma companies, and that was licensed, it was patented, and we screened so many products. You name the companies, any big company you talk about, we have tested their products and given them the data to shortlist. Because clinical trials are expensive. If somebody can tell you what are the 3 likely molecules or formulation variants which are going to work, it's the best thing you could do, because you don't want to take all 3 candidates to the trial. So it helps in candidate selection. And this work I did in Huddersfield, which my PhD student Joao, and what we did in this project and this PhD, we wanted to make a simple laboratory QC method to identify how these polymers ionize. And guess what? We just used Malvern's synthesizer, which every company had it. And we developed this method for industries to then find out how their tablets are behaving, how their coatings are dissolving. And we published this in open access, and it was a big success. Bristol Myers Squibb on the phone next day saying, Hamid, can you run 10 samples for us in Huddersfield? Yes. So they've got their own manufactured polymers, they grafted them, and they said, well, this technique will tell us something which nobody else can tell. So they sent us samples, we sent them data, and then they were very pleased that they could now identify the polymers which will work best for their candidates, which they were formulating. So again, open knowledge, published it. Some pharma companies are benefiting from it, and why not Pakistan? Another example was this duo coat system. This was again a project in UCL I did. And this idea was that you double coat a product. Now people will say, well, double coat it will gonna take long time to dissolve. But the inner coat was cleverly designed to dissolve outer coat. So the outer coat sometimes struggled dissolving because of the environment in the small intestine. So what we did was we put an inner coat which eats the coat from inside, and the gut fluid then eats the coat from outside. So it dissolves faster. And this duo coat system was then out licensed by a Swiss company called Tillot's, and they had their new product just launched in 2019 using this technology, and their benefit was faster release after gastric emptying. So after gastric emptying, it could take 2 hours for products to start dissolving. And if it doesn't dissolve, it ends up in colon. Colon do not have enough water. It comes out in the toilet. So lots of clinical trials showing patient coming to gastroenterology clinic saying, look, tablet comes out in the toilet. It doesn't work. It's not the poor quality tablet. Tablet was fine. It's just the physiology. Sometimes they don't have the right trigger. So polymer will only dissolve if the pH in the gut goes above that threshold. If the patient has very active disease, bleeding, actively ulcerated, inflamed, it's very highly acidic, polymer not going to dissolve, tablet going to come out. Same happens with Epival. We used to get lots of complaints from patients saying, I've got seizures, episodes, your product is rubbish. I had a vomit, product came out intact 3 hours after I took it. It's not working, bloody hell, what did you put on tablet? And we were like, this is the quality of the tablet, that it did not dissolve for 3 hours and it resisted your stomach. Because your product was never emptied from the stomach, it's not going to dissolve. But the point is that if we do not formulate the product based on the physiology, we cannot improve the clinical efficacy. So my message to you is stop copying what somebody has made, and slight change in the formulation can improve clinical efficacy, and that's where the innovation lies. Very cheap innovation. You don't have to rediscover things. Within the generic industry you could do that, and you could demonstrate the benefit in a very small clinical trial. Even it's not a new product, it's not a new molecule, you just have to demonstrate the benefit, then clinicians will be with you. They will say, oh yes, you're right, it works, it's better than that product, kind of thing. So this one is out licensed. Then I made this one. So this is Huddersfield work. Again, one of my PhD students did it. And this is about the capsules. So I was talking about a company in Punjab making capsule shells. So what we did, we said, look, you make enteric coated tablets. So you make a tablet and then you have to go through coating. So if the coating has got pinholes, it has got problems, then you are responsible. You have to do the QC. So how about you make a capsule which is inbuilt enteric? So you buy a capsule, fill it in, package, go away, saying, all right, anything goes wrong, capsule shell manufacturer is responsible. They have to do the QC. How do you do that? You can't coat a capsule because it's a poor strategy. Lots of people tried. It doesn't work on mass scale production. Okay, so never coat a capsule. Doesn't work. So what we wanted was to have enteric properties built inside the shell. So don't coat it. You buy a shell, open it, fill it, close it, package it. That's it. So we did lots of initial scientific experiments. It failed, and Evonik was trying it as well. Evonik was one of the biggest supplier of Mamaco polymer, methacrylate polymer, enteric polymers. It's the biggest German company who holds the market share for enteric polymers. They are the innovators in polymer industry, if you like Dow Chemicals. So we did this, and we published our paper. Next week, guess what? Evonik's director of R&D was in London talking to me, saying, how did you do it? We've been trying to do it for so long. How did you do it? And so that's the capsule we made. That's the capsule, and that's the release profile. So what we did was we made capsules with different polymers, and we achieved variety of different profiles. So what we said to Evonik is like, look, you can tailor the drug release by changing the composition in the capsule. So you can have a capsule if you want to target upper part of the small intestinal tract, or the proximal small intestine, or jejunum, or you want to go to the colonic region. By just changing the composition of the polymer A and B in the capsule shell, you can achieve a variety of profiles. And they were very interested. And you might hear a new product launch in next year. Then what? Everybody gonna, either ask for licensing or trying to copy it, but we can innovate. It's not difficult. What we did was just change the composition in the gelatin mixture, which is used to make the capsule. And that works. So again, thinking out of the box, coming up with new ideas which are easy, does not require toxicology, does not require phase 1, 2, 3, 4. We just need a small clinical trial to show that it works and it's better. And it's not very costly. And if you do that, if you scientifically prove it, regulator will convince, they're not going to argue with the evidence. But because we do not produce evidence, then they say, bring evidence from outside. Has anybody else done it? And of course, they wouldn't have done it because you have done it for the first time. So this demotivates the industry to innovate in Pakistan because nobody else has done it, so you can't do it. But that's not true. You can do it, and if you provide the evidence, regulator will be fine. But what we do is we make things, we change things, and we don't produce evidence that it's safe and effective. That's where the regulator becomes uncomfortable because they're taking responsibility on their shoulder. So they will ask you, well, no, do something which Canada has done, do something which US has done, do something which UK has done, so we are safer in your strategy. Now, this is another interesting product I worked in UCL. And this was for ulcerative colitis. So we use a different coating. This is a starch-based coating. Now imagine starch, how cheap it is. So we use starch, just maize starch. But maize starch, people can't digest properly. So some part of the starch goes undigested in the colon. And then the bacteria eats on it. So it's a fermentation product. So it gets digested ultimately at the end, but your body's enzyme doesn't digest it. It's the bacterial enzymes in the colon, so your microbiota. So we mixed the starch in acrylic polymer coating, and we developed this coating system for ulcerative colitis, and we did some gamma scintigraphy clinical trials. So we found that the traditional tablets of Asacol, for example, with mesalazine for ulcerative colitis, lots of tablets came out in the stool undissolved, and our product all dissolved in the ascending and transverse colon. There was no clinical failure. So we had a discussion with a gastroenteric company in Switzerland, Tillet's Pharma, and they said, okay, that's our next generation Asacol. Thank you. So they took it. The clinical trial was conducted in 2018, and this took 10 years for us to work on it. And 2019 product launched in Europe, and 2020 product launched in US. All made in university, out licensed to a pharma company. And this is not really very difficult. It was just a change in formulation. It was nothing to do with API. It still contains same 5-amino acetyl salicylic acid, mesalazine, same API, nothing new. So that's been launched. And that's the press release of the UCL at that time. They were very proud that the university's product is now going out in the clinic. Now this is what I did in, again, Huddersfield. And what I did was, because I've been working on coatings, and I was like, nutraceutical industry is always in trouble because you've got these garlic tablets, you've got all these special nutraceutical ingredients, but when you take them, the stomach destroys everything. So it doesn't work, either chemically damaged, or if it doesn't get damaged, you get nausea and bad breath, so people don't want to take it. But nutraceutical products are not tested for clinical trials, so they are sold in the market without efficacy testing. So how we can improve the delivery of those products? So I wanted to have a coating which is pharmaceutical standard but can be used in nutraceutical. But you would ask the question, why pharma coatings are not used in nutraceutical? Because if you put the pharma coating in, the government would not allow it because it's not nutritional based, it's a synthetic polymer. So once you use a synthetic coating on, you can't sell your nutraceutical without MHRA getting involved. So lots of companies knows that their product doesn't work, but they don't want to put a coating on because that will mean that they will not get a license. So what we wanted to design a coating which is again based from natural ingredient. So there were some natural ingredients which we could have used, but there was no patent on it. So pharma companies wouldn't be interested in it because there's no return on investment. So I was fishing for polymers which had never been used, looking at the chemistry, and I found one from a gum, natural gum, which is amazingly available across the world. It's available as a food polymer, so it's cheaper, available in a good quality, already used in food production, and we used it in coating application, and we had filed a patent 2 years ago, and we're working with a pharma company to get that product out in the market. The beauty of that coating is that it's an entire coating, but completely natural ingredient from a gum, from a plant-based gum. So you can apply it on all those garlic products and all those nutraceutical products, and they will be delivered in small intestine instead of stomach. So that's available for licensing to other applications. Another one I worked again from nature. So when my first daughter was born, she struggled with childhood eczema. And atopic dermatitis in United Kingdom, babies grow out of it. But at that time, as a young parent, without any social support, my wife and I were struggling with first child, with eczema. It was a very difficult quality of life. The baby would not sleep, she had allergy issues, she would wake up in night, and I will put her on my shoulder, walk around whole night, and my wife and I will take turns. So I was like, bloody hell, what should I do? The doctor prescribes steroids, but I can't use steroids every day because it's obviously had long-term issues. So I use steroids only on chronic flares, but I want to use something else. So when I look for something else, there's emollients, but emollients doesn't work. You again get a flare up, you use steroid, you keep using emollients, all those range, nothing works. So I started looking for a product which is not steroid, but not an emollient, something in the middle which works. And I found a particular extract from a plant which worked amazingly great. So I started a PhD project on it, and guess what? At the end, we had a product now which has been trialed with the pharma industry, and it's a herbal product, so it doesn't have to go through all those clinical trials which you need for a pharma product. And this might come out soon. And this is a topical product you apply on the skin. It's a plant extract, and the beauty is you don't have to go in Africa and cut the trees and extract the plant. The extract is available as a BPC Codex product. So you just basically buy it, formulate it as a cream, here you go. Easy innovation, right? Because if you go about saying, okay, this is the tree, you're going to cut it, you need now extract, standardization, it's not in pharmacopoeia, they're not going to approve it, right? This is something easy. It's a BBC grade product, traditionally used for burns and cuts, never used for eczema or atopic dermatitis. And the evidence we have is interleukin 6, interleukin 12, TNF alpha, with amazing pharmacology. You will be shocked. And all the evidence we had is a cultured keratinocyte. So what we did was, we cultured the skin cells keratinocytes, which produce inflammatory cascade on a dish. And we did all the trials and proven that it works. The efficacy is equal to a steroid. A very good product. Again, coming out like this from university. So moving forward, we need to transition from classic generics to high value products in Pakistan. If we don't do it now, we won't survive longer. There's huge competition. We have got hundreds of manufacturers in Pakistan. Slowly, industry will start dying. So we need to survive. The fittest will survive. The companies who will take initiative now will survive. So diversify your portfolio, move away from classic traditional products, invest in new technologies, or come up with innovations within the generic sectors so you have some niche products coming out. So reposition the export market so you can sell something which nobody else is selling. So again, people will buy from Pakistan. And there are rooms for improvement when it comes to Pakistan. See, I was on the shop floor, so I can tell you what issues we have. So make sure that the pharma companies in Pakistan try to aspire pharmacopoeial compliance. There are still majority of products which do not comply to pharmacopoeial standards. And it's not just local companies, even with some good names. The APIs and formulations don't comply compendial specifications in full. And that's a big issue. So this will eat your exports, this is going to cause you huge problems and reputational damage. So please try an internal project, something maybe 5 years project, maybe 10 years project, but put it something on place saying, look, you are the plant manager, you're the QC manager, have a pharmacopoeial compliance in 5 years. Give us the gap analysis. Where are we lacking? Why, what areas we are not meeting and what we need to meet those compliance. And I'm very sad to say that majority of products do not comply. And this will restrict our export market. And not only that, but reputational damage. So impurity and related products is the biggest problem. We do not test impurities and related compounds to the level required in pharmacopoeia. And that is a big issue. It's not a problem for products like paracetamol and ibuprofen. But think about antipsychotics, think about antibiotics, think about steroids, think about those niche molecules and injectables, the products intrathecally taken in aesthetics. It's important. And if you don't do it, it's not known, but once it's out, it causes huge reputational damage. The other biggest area is the bioequivalence. So lack of bioequivalence is now eating our exports. So countries like Jordan, countries like Syria, small countries which never asked about these things, they are demanding bioequivalence. So when are you going to do it if you don't do it today? We have center of bioequivalence now in many cities. We have some universities run centers. We have some hospital run centers. It's not expensive, but it tells you what your product is capable of. And I know when you do the bioequivalence, your product will fail. Majority of product will fail. So you should have a plan in mind that how would you reformulate to make it bioequivalent. And my suggestion would be do an in vitro bioequivalence first without going for an in vivo bioequivalence, because this will tell you where you're standing. So you can reformulate, do a QC again, and then put the best product out for bioequivalence. But once you have a product, please don't change it irrationally. So your materials manager would come in saying, look, this material is short, I need to buy it from this company. You change the source, you ruin your bioequivalence completely. So not many companies do vendor approvals, not many companies do compliance, and this is our biggest reputational damage in the world. And people know about it, which is why people are asking questions on poor quality generics. Generics, as Professor Zahir said, is an important part of the supply chain. WHO loves it. Everybody loves it. We can't survive without generics. They're very good. But if we don't make good quality generics, we can't change people's perception. And once a reputational damage happens, then it kills everything. So you remember the issue with the Lahore case, the Punjab Institute of Cardiology. You would not anticipate how much reputational damage happened on that occasion. It was really appalling. I was in University of College London at that time, and the samples came to our lab for testing when the MHRA was investigating it, and I was literally crying, thinking what's happening. A mistake can happen, but covering up a mistake is an offense. And the reputational damage happened because we covered it up. So we didn't raise our hands up, say, look, this is a mistake, sorry. We didn't do it intentionally, this is what happened. We didn't do that, we started covering it up. And that caused a huge damage to our exports. The people stopped trusting Pakistan, and then, the consequences are very severe. This is a burning issue now in Europe. So you might have heard about some people who are sitting from the QC side, they would know about the nitrosamine impurity issue. So we had a huge shortage of sartans, losartans, irbesartans, and ARB blockers, antihypertensive drugs, just because of nitrosamine impurity. So the biggest API manufacturer in China voluntarily reported to European medicine regulators saying, look, we have got this stability issue in our product. And when they reported it, oh my God, EMA was shattered, absolutely shattered what to do. It was a carcinogenic impurity. But what to do now? It's present in bloody hell every other product. And that supplier was the highest trusted supplier. It was a very good product. And lots of multinationals used to buy from that. And there was a huge recall, and I remember Pakistan rep also initiated a recall. Sartans were out of the market completely. But then what did we do? We're selling it again in Pakistan, aren't we? But think about what EMA did. They had a roadmap. They've asked pharmaceutical companies to control the level of impurities to the bottom. So they've got PPM limits set up, and they said, okay, let's do the risk analysis, saying, all right, if you've got this much levels in your product, people take one tablet every day for 10 days, how much exposure they will have, and if they don't have an antihypertensive tablet, how much damage they will have to their system. So they did the analysis, they allowed some bad products for few months in anticipation that companies will meet the targets in due course. So they've given them 5 years timeline. How beautiful is that? They didn't say, okay, take away all the products, nobody going to use it. What hypertensive people going to do? People will have other severe consequences, not having a product at all available. But the beautiful strategy was they worked with the industry, they give them a timeline saying, okay, within 6 months you do that, within a year you do that. If you don't do that, your product will be out completely. You can't sell it. And the companies will move on. And guess what? Last month I saw a publication where somebody cleverly reformulated the tablet and they added an excipient in the tablet which reacted with the nitrosamine, so the API don't get react with that. So they provided a scapegoat, like something to sacrifice as an excipient. So that product gets saved. The API is now protected. So solve the problem. But think about Pakistan. What did we do? We did nothing. And this problem then rolled out to ranitidine, famotidine. There are 15 APIs affected with nitrosamine impurity, which is highly carcinogenic. But do we have a roadmap? No, we haven't done anything. So we just recalled some batch and then we went back. All right, let's buy it again. And China would have probably dumped all the bad ones to us because EMA wouldn't buy the previously specified product. So they have got new specification now. So what those companies going to do? They have no mercy. They're going to sell it to somebody who don't care. So think about it as a responsible manufacturer in Pakistan, see where we stand and what we can do in our capacity to make sure that we are still the best generic manufacturing country in the world. This was a very eye-opening story from GSK. I don't know how many people know about it. This is a bit 10 years old, but GSK was accused in China for unethical marketing and bribing doctors. So they were fined millions of pounds, and the CEO was imprisoned, he was sentenced, and then he was deported from the country. It was very appalling, shameful incident. He came back to UK, he was a British citizen. But think about Mark Rayleigh has gone through. What they were trying to do is completely appalling. So when Mark Rayleigh was interviewed by BBC in London, asking, what did you do? How could you do it? And he was like, look, I will be honest. This was always how it was done in China. But we need to understand that ethics are improving and countries are changing. The ethical dynamics are changing. So we can no longer do what we did in the past. And this was the biggest lesson a big company learned. But what we are doing in Pakistan, again, following the same trend. So this is a wake-up point, for our improvement, that what we can do by learning from these lessons and not repeating what happened in Punjab Institute of Cardiology incidents. It kills all the reputation. And we already had one incident. Another example I give you is from India. It's again a huge issue. Ranbaxy. How many people know about Ranbaxy? I don't know. But what happened with Ranbaxy? They were falsifying data with FDA. They kept falsifying it, and they provided clinical trial data, and that trial never happened. All blood samples were completely fake. And the Ranbaxy's director in US found out, and he reported to FDA. And you can think about the consequence. It was a very long investigation, took about 7 to 8 years. At the end, the company was completely busted, taken over by Sun Pharma afterwards. And reputational damage to India, humongous. People stopped trusting, saying, oh, bloody hell, we don't trust them anymore. They faked so much. So there was a whole faking machinery. They employed hundreds of people to fake the data. So people think, oh, if you could spend that much money to fake it, why can't you just do it? It's the psychology, just fix it, do a shortcut kind of thing. So huge reputational damage. Please, be aware that it's not just the company, it's the national reputation on stake as well. It's not just you getting affected, all pharmaceutical industry gets, affected. So I wrote an article in Dawn a while ago about the generic products and 10 reasons why our generics are not to that level. And I criticize some of the new regulations coming up with generic prescribing. So if you like reading it, there's a link down there. It's written in a very easy to learn, understand language because it was for a newspaper. But this will give you some key burning issues from a scientific point of view, that what's the industry lacking in Pakistan, how we can go from A to B. So lots of things are unknown unknowns. I'm not saying everybody is cheating. No, that's not what I'm saying, because I worked in the same country, in the same company. There are lots of things which are unknown unknowns. But once they are known, it's our ethical responsibility to sort them out and have a plan in place to fix it in a period of time. So don't say you fix it tomorrow. At least you should have a 5 years plan, 10 years plan to go from A to B. And then you will be aspiring to the high quality generic producing company or a country as a whole. So ethics, regulatory compliance, and better governance are the key to success. And that's where I conclude my talk today. If there is any question, I'm very happy to take. So 4:30, half an hour before, right on time.
I think the industry stalwart sitting around this hall or in this hall would see that there are the opportunities. There are more opportunities than the challenges, which you have highlighted. I think you have given us food for thought, and as an industry, we need to change our practices. We don't want to be Ranbaxy of the world. Uh, let's give him a big applause, please, once again. Questions, please.
Nice to see you here. After such a long time in Abbott, we were together, and I'm very happy. And I got very good information out of your work. I would like to ask a question.
Sure.
That you have said that you have worked on fluoro tablets. I only want to, some purpose, I want to ask that more related names of tablets. Can you tell me which have you worked in related this fluoro tablets?
Which tablets? Can you say it again, please?
Fluoroguanol tablets.
Fluorobutanol
Yeah.
tablet. No, I didn't work on fluorobutanol.
Okay. Okay. So thank you very much, Hamid. for such a-
Ulcerative colitis. Okay. Are you talking about
Yeah.
ulcerative colitis?
Yes.
So what we were trying to do is, the ulcerative colitis is a condition of colon where colonic mucosa is inflamed. So you have an inflammatory bowel disease which is confined to the colonic regions. You call it UC, ulcerative colitis. There is another condition called Crohn's disease. In Crohn's, you have inflammation all across the GI tract. So you don't have a specific region. So you called it Crohn's. In Crohn's, you have more immune pathology along with inflammation compared to UC, which is confined to inflammation only. So what we were trying to do is look at the disease pathology, and we found majority of products do not work just because the drug do not go to the local tissues. And in the earliest stage, Crohn's or the ulcerative colitis is not a systemic disease, it's a local disease. Say for example, if you got a wound there, but you apply ointment on this hand, would it work? It wouldn't work. If there's a wound here, you have to apply the product there. Similarly, in UC, you have an internal wound and you want to apply the product on the internal wound. But how are you going to do it? So some clever gastroenterologists used to use endoscopy. They used to go down, spray it. They used to use very innovative surgical tools to try to remove or heal the inflamed tissue. But you can't do it every day. You can only do it on acute conditions, maybe once in a year. So what we wanted was a product which goes down and target the colonic region to cure or mediate the inflammation. So we focused more on formulation strategies rather than API because we believed that the drug works. Even aminosalicylic acid works. It doesn't work because you're applying on the wrong hand, for example. So if the anti-inflammatory goes to the tissue, you will get a response. So we did it with budesonide steroid, we did it with 5-ASA, and we did it with some immunosuppressants as well, and it works. So the coating technology is licensed by Tillotts in Switzerland, and they now own the rights for all the GI applications. But then we have licensed it for some other companies for colonic absorptions like the statin project, which you mentioned in the introduction. So technology is available for any drug which you want to be delivered in the colon and for whatever application. So for statin, we wanted to just avoid gut wall metabolism. So say 10 milligram will work like 60 milligram, just because you deliver it at the place where it would not metabolize. So efficacy is 6 times higher by just putting a different coating. So how beautiful is that? So we invest a lot of money in developing new molecules, but sometimes products don't work because they're not formulated better. They're rubbish. And pharma companies, when they design new molecule, they spend huge amount of money, 7-8 years to discover the molecule, do the toxicology. So when they eat their patent shelf life, they're in a rush to launch the product. So they do not spend too much time in formulating. They launch it very quickly because their patent is finishing. And then in parallel, they start the formulation project. And as soon as the patent expires, they bring a better product out there, get another shelf life for 10 years afterwards. And if you don't do that, they won't going to get a return on investment on the discovery. But because you are generic companies, you're not investing on R&D, you're not making new molecules, you could do that early on, you know. So sometimes people focus so much on API, they forget the excipients. Trust me, excipients are as important as APIs. I haven't got the slide on that, but in another project in London, we were doing some work on excipients, and we worked on polyethylene glycols, we worked on lactose, we worked on some of the coating materials, and we found that for class 2 BCS compounds, like with low solubility and poor permeability, what happens that excipients increases the bioavailability in males and they decrease the bioavailability in females. So it's a gender effect. And then we checked it with 5 other compounds, and this is third PhD student working in this area now consecutively. And FDA is worried, and the Center for Drug Research in Women is getting involved, and they're now trying to find out why it's happening. And they're talking about blue pills and pink pills, some products for males and females separately formulated just because of that. So formulation is so important. You know, it will make a molecule ineffective completely if it's poorly formulated. And a very old drug will work miraculously great if you reformulate it nicely.
Thank you very much. Thank you.
Any other questions? Yes, Miss Maria.
Good afternoon. I have 2 questions. The first question I would like to know, are the vaccines and serums considered as biosimilars? And number 2 question is, on the drug development perspective, is there a need to do biosimilarity tests for vaccines and serum preparations? Thank you.
It's a very tricky question. Normally, biological products are required to be tested as biosimilars, but vaccines are tricky products because they are different. Each product is different. And as far as you have immunogenicity and clinical efficacy data, you have your own product. So it doesn't have to be bioequivalent like biosimilars. So there's a new term now being used in the US called biobetters. So in the past, because of biosimilar regulations, the generic copy has to be as bad as the innovator one. So not only as good as innovator, but also has to be as bad as innovator one. So you can't make it a better product because biosimilars has to be same. So the reason was that if you make it better, then patient can't switch from product A to B because the efficacy will change. You have to re-titre the dose. So the new term now coming up from FDA saying biobetters. So you can make it better, but then they will be rebranded and then patient can't switch over. So it depends on how regulatory bodies see that product and what data set you have got available for your product. So if you have got a vaccine where you have done all the way from immunogenicity trial to efficacy trial, you don't have to show biosimilarity, you just show clinical efficacy. But if you don't want to do the test, you don't want to do immunogenicity test, so then you will have to do biosimilarity because otherwise how we would know your product will work. So it depends really what data set you have got, and different regulators will have different preferences, for example. So say if you go in the US, they would not like biosimilarity on vaccine, they would want clinical efficacy data. So it's a choice of regulatory body.
Pakistan pharma industry, you have worked in here long time now with the global perspective. What one thing or 2 or 3 things you would suggest we should do and excel in that?
Thank you, Harun साहब. I slides दोबारा से ऑन कीजिएगा प्लीज. जी, आपके आने से पहले कंक्लूडिंग स्लाइड मैंने यही बनाई थी, तो आपको दोबारा से दिखा देता हूँ. तो what I would suggest is that we transition from classic manufacturing to innovative products. If you don't do it now, my perspective is it would be too late. I had some experience working with Nigerian government recently and with Jordan government and some of the Middle East. Some countries are stepping forward, and Pakistan has always dominated this region in terms of exports, and our products were most beloved in those regions. But we are now losing our share with time. So if we do not change, somebody else will come in. Now there are problems working with China and India, so not everybody would like to work with those two countries. So Pakistan is a very good alternative. So Pakistan has hundreds of pharmaceutical companies, and many would not survive. So fittest will survive. So investing in the right technologies. In previous slides, I've given some suggestions in terms of what products we can think of, not just products, the areas, and how those platform technologies can be invested in multiple dimensions. So those areas, and also I talked about these three things which I personally feel that Pakistani pharma companies lack. One is pharmaceutical compliance, the other one is impurity and related compounds on stability testing, and third one is the bioequivalence, because now everybody going to demand that. You would remember Syria, Jordan never asked for these, Nigeria never asked for these. Now I was sitting with Director General of Nigerian FDA, and she said, I'm not going to allow any product without bioequivalence in my country. Now this is how countries are changing. So this is my suggestion, take home message for the pharma companies, that you act now. Those people who will cleverly invest today will benefit 10 years later, kind of thing. And also the reputational damage was the other thing I was talking, that if there is something unknown, okay, we didn't do it purposely, but if we found out something is wrong, it's our ethical responsibility to take the ownership and say, look, sorry, this is what happened, we didn't do it intentionally, but this is what happened. But if you try to cover it up, it causes more reputational damage. So we think about, oh, if we don't talk about it, it's going to be fine, but it pops out. Then I mentioned about the GSK incidents, which caused a big shame of the British Mark Rayleigh. When he came back to London, he was really struggling with the reputation. And then I talked about Ranbaxy's case, which caused a huge problem in India. And this is something which ruined Indian reputation in America. Now nobody's trusting their products after that happened. And we don't want that for Pakistan. So we had one incident, and we don't want another one. Then I did talk about this recent issue about the nitrosamine impurities, which affected a lot of molecules, started with sartans, but then it rolled out to many other APIs. There are about 20 different drugs which are now being affected, and EMA had a rolling schedule for pharma companies to comply, but Pakistan didn't have that. So my concern is that obviously people don't know about it, but they will find out. Once they find out, it's again a huge reputational damage because it's a carcinogenic impurity. The other problem is that Chinese manufacturers have no mercy. So if Europe is not buying it, they would have shoved all the stuff to Pakistan and African countries. They're not going to waste it. So if you are not clever enough to pick it up, we are unintentionally getting into this issue. But at the end, from regulatory point of view, they will blame the manufacturer. They would blame you, saying, why didn't you check? So you can't blame Chinese companies saying they supplied it. They say, why didn't you have your internal control? Because DREP already issued a notice 3 years ago. So DREP not going to take responsibility either. So I ended my talk with this note that ethics, regulatory compliance, and better governance are the key to success for the Pakistani pharmaceutical industry. So that was a quick recap and answer to your question. Any other question? Yes, our staff. Mike, please.
First of all, a very good talk, Hamid. It's really a pleasure seeing, because Hamid and I we have been together for the last 25, 26 years. Our friendship goes that long back, and it's really a pleasure seeing you over here, growing so much, doing so much of an innovative work. I have few reservations about some of the suggestions that you have put forward, and I think I would be rather advocating for the industry, because what you are talking about and what you concluded on in terms of better governance, I think how we actually translate that governance into practice and actions is really the point that needs to be considered and talked about. And I would refer to what happened last year when we were getting the global trials of the global pharmaceuticals and the vaccine manufacturers for the COVID vaccines. And our bioethics committee, the national committee, actually stopped some of those incoming advancements in technology transfers. With some very, I wouldn't go into detail because industry stalwarts are sitting over here. They would know what was all the fiasco about. But I think governance shouldn't be about strangulating the neck of the industry in that way. In that manner, I would advocate for the industry as well as the academia, because if you wanna grow that culture of innovation, if you talk about bioequivalence in clinical trials, we need to be receptive, because we are not doing anything innovative on our own. So first of all, we need to import that technology, that knowledge, and then perhaps we can think about growing on our own. But if we stop that entry and we stop that transfer, how do we grow then? So
No, Aftab,
in
you're
that
right.
case, I think
You're-
what
yeah.
Kaisar Saab was saying earlier,
Right.
that regulator needs to play a positive role. That needs to be underscored, I believe.
No, I do agree with you, and I wouldn't disagree just on the sake of disagreement. But governance comes on the regulatory side. So it's not a burden on pharma companies. Pharma companies has ethics and compliance, and governance comes from the trap side. So there's a poor governance from the government side as well. So I don't want to just conclude on pharma industry. My conclusion was overall saying we need a better governance from the DREP side as well, because they're not good as well. They've got a lot of issues. They've got poor understanding. They go by very orthodox traditional methods of regulatory governance. So we do need better governance from their side. I didn't mean that it's the governance from the industry side. Yes, sir.
Hello, Doctor. The biggest killer in Pakistan, and 35 million people are suffering or at the high risk of diabetes. And diabetes cannot be curable but can be controlled. So why don't you suggest some kind of drug manufacturing for that disease? And because this disease is because of our cultural values, or we eat so much sugar and like that, or you can suggest some kind of medicines or developing of some kind of pharmaceutical.
It's a good comment. This reminds me of a beautiful phrase from a poet, and he said, "बड़े बेआबरू होकर तेरे कूचे से हम निकले." But if you look at the first line says, "निकलना खुल्द का आदम का सुनते आए हैं लेकिन." So that wheat is a problem. ये जो wheat है, and diabetes is an issue, is an overall issue of our culture, the society, and over reliance on a bread. So the new guidelines are coming out on a low carbohydrate regime, because we in Pakistan and some South Asian countries, we consume too much carbohydrate and starchy carbohydrates, and those starchy carbohydrates converts into a reservoir, cause more insulin resistance, and hence the diabetes burden. So hopefully the nutrition societies in UK have been debating this for 10 years, because NHS would go on low fat diet, and now the new recommendation are saying you can have a bit of good fat. For example, you can have a full cream milk or a semi-skimmed. You don't have to go for a red one, for example, and have some good fat, some nuts, some Mediterranean diet, but go down on carbohydrates. Because what happened in UK, everybody loves baking. So when people go on low fat, they start eating cakes and pastries and bread and rice and all that starchy stuff, and that causes another burden on the other side. So it's now been scientifically agreed in United Kingdom. From regulatory point of view, it will be rolled out soon from NHS. So they will be advising patients to go on a low carb balanced diet rather than a low fat diet. And I think in Pakistan we do the same. So जब हम खाना खाते हैं तो सालन कम खाते हैं, रोटी ज़्यादा खाते हैं, रोटी से हम पेट भरते हैं. What we need to do is we have to eat the food, not the bread. Bit of a bread, fine, but we need to eat the food. And ये cultural change इतनी आसानी से नहीं होगा. And we eat lots of starchy products, potatoes, rice, and all those underground vegetables, and they are a huge reservoir. But with time, in the past it was okay because we were agricultural country. We used to work hard, हम मेहनत करते, बहुत काम करते थे, दिन रात मेहनत मजदूरी करते थे, सब burn हो जाता था. Now we are more intelligent. We have a knowledge economy. We sit on, work from our brains, but we eat a lot, so we don't burn enough carbohydrates as we used to do before. And that's what's causing this pandemic of diabetes, in my opinion. It's a pandemic of insulin resistance, and that shifts the balance towards cardiovascular disease after. When insulin levels are high, because what happens when when your blood sugar is not controlled, your pancreas produces more. So in the first stage, you have hyperinsulinemia, which is very high circulatory insulin, but you have no hyperglycemia. You're fine, your blood sugar is perfect, but your insulin levels are 10 times higher. Then the pancreas get damaged. You get starting on insulin products afterwards, but higher insulin levels are very dangerous for arteries. Insulin is a trafficking hormone. It causes channeling of calcium, so that causes your artery damage, which causes cardiovascular disease afterwards. So diabetes takes you on that road of cardiovascular disease afterwards. And then we talk about low fat diets. Yes, low fat has a problem, cholesterol has a problem in atherosclerosis, but the underlying cause is insulin. So we have to retwean our body and reduce insulin resistance in the muscles and the adipose tissues. So if our body increases the sensitivity to the insulin, we better use insulin, then we don't need too much insulin. So insulin level goes down, and then the arteries are happy.
Yeah, one more question, if you don't mind. I mean, in Karachi, many people are using subsoil water, and that because of that, they have the skin problems and eczema like that. And eczema again is not curable, but I think fusidin H will be helping people like this, or other molecule like that. Eczema.
Can you explain what subsoil water?
Subsoil underground water that is pumped out, and people are using that because of the scarcity of the water, because of that itching problem and like that. And
it depends really, because I don't know about an evidence that how water could relate to eczema. But say if water is contaminated with allergens, which your body would react afterwards, it is possible. But I don't know if we can generalize this to every kind of water from the well source. Some water might be highly purified and will have no problems, but if the soil around the area is contaminated, or there's an industrial waste dumped into that area, and the water you suck out has got a lot of contaminants or subclinical doses of chemicals and drugs or byproducts, then definitely you might react. You will have got huge immunological reaction to that.
Is it drugs for that?
It's the water governance again in Karachi. Unfortunately, we don't even have water. We buy water and pay water tax as well. So what can I say?
Then the solution could be the fusidin H like drugs or that for the eczema.
I don't think we need to treat the disease. Say this is my argument for typhoid and cholera as well. I always say people go, oh, multi-resistant, we need this antibiotic combination, we need 2 extra vaccines and da 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 da. It's a waterborne disease. You provide a clean water, that disease will not exist. So we have to fix the cause. We are treating symptoms. We know this comes from poor water, so we need to provide clean water, and the city governments and the provincial governments need to invest on those areas. What's the point spending $1 billion on a new multi-resistant vaccine instead of putting $1 billion project on the clean water sanitation? I don't understand that. Plus, if you look at the public health data, so people would say, look, hospitals are full of multi-resistant typhoid cases. But if you look at the catchments, those patients come from districts where clean water is not available, but you try to vaccinate urban population who is drinking mineral water. There is no correlation with that. But with NIH, what we could do is we can map the data. Now, this is where pharma companies could come in. They can map the data saying, okay, civil hospital has got this much patient, Jinnah hospital got this, private sector got these, where these patients are coming from, what's their postcodes, which catchment areas they're coming from, and then do a heat map and see what's causing it. And then we intervene accordingly. This is where the public health governance come in. But again, public health in Pakistan is not like PHE in England. So we don't have that. If pharma company don't have that, they can't benefit. So unfortunately, we need a different strategy. We can't have public health England in Pakistan. So pharma companies might want to work with some public health academicians, some clinicians to generate that data source and then direct their drugs marketing plan accordingly. So I have a very different view. I'm not against vaccines, but I am a advocate for rational use of medications. So we do not in UK promote irrational use of antibiotics and vaccines and medications. We always say you take what's required and not unnecessarily take products which can be resolved by alternative means. For example, the principle of vaccine is prevent then cure. Same thing goes around here, that even if you can prevent a vaccine by better water, then you do that, kind of thing. Yes, at the back.
Hi, thank you for the great presentation. I'm from Swiss Pharma, and I have a question regarding to your presentation. You mentioned about ibuprofen in your slides, and you also mentioned that excipients are also as important as the active pharmaceutical ingredient. So my question is, in Pakistan, obviously we are a regulated industry. Do you think we have the infrastructure for R&D, especially which is backed by the government, to guide and inspect on the excipient side? Because most of the pharma companies are, mostly centralized on the active pharmaceutical side. So do you think, obviously for a developing nation like UK or US, they do have a wide R&D department at the government level, but what about the case for Pakistan?
It's a good question. Lots of people ask that question, but to be honest, it doesn't need huge money. Excipients can be tested simply in the lab, and a normal QC lab can do that. But because we have a perception of excipients are inert, they have no role in the product, so we don't focus on those. So you can use your conventional techniques like HPLC, UV spec, dissolution test, XRD. You can use Raman spectrophotometer or FTIR, and you can do some basic data analysis, thinking how excipients are interfering with your product. And you can also run some quick stability samples. So your routine batches can be left on stability for learning, not just because regulatory wants it. You can put some batches on and see how change in excipients affecting your product down the line, what you made change, how does it resulted in, say, 2 years down the line, and what change B looked like, and learn from that experience and modify it. So you don't have to invent something. It's a routine learning. It's an R&D by going with the production by your QC analysis. So I don't think it requires a huge infrastructure. Sometimes you do need some electron microscopy or some technique which you might not have, but universities have it in Pakistan. You will be surprised how very well kitted some universities are. They never use their equipments because they don't know the applications. So you can go to them saying, look, you've got this machine. Can your university student work on that project for us? The students are doing rubbish project. You can give them a project, supervise it as well. The lecturer will be so happy because they don't have to supervise it. 1, 2, they don't have to think about the project, and your work will be done for free. So there are lots of opportunities, and I say it by example because I do lots of projects in my lab which I don't charge, because they're exploratory projects. We do it with companies sometimes, and sometimes the money is being given from somewhere else. So if we can do it as a science, as a learning, we do it. But sometimes if it's a pure consultancy work, we charge for it. But universities in Pakistan, if you give the cost, they will be fine. If you give them the material, give them the reagents, university will be so happy. They're not going to ask for a remuneration on the top. We do sometimes ask, but I think the Pakistani universities, the professors, the academics are looking for working with industry. Lots of people are very keen. But because we don't connect with each other, and we think academia do bad research, and academia think industry never wants us, so we never marry together. But if you approach them, they will be very welcoming. They will open their doors. If one university doesn't want it, you can go to the next one. I've been told in Pakistan, when I left many years ago, there were only 4 or 5 institutions teaching pharmacy. Now there are 20, probably only in Karachi. So there is a huge potential.
Thank you, Dr. Hamid and Zahir Saab. Dr. Saab, बहुत शुक्रिया आप लोग गाए. Let's give them a very big hand. As sometime Englishman says that yesterday I was wise only. Yesterday I was wise only. After listening to today's thought-provoking lectures and presentation, I'm going to be a wiser person. You're going to say that tomorrow. Okay, you are now wiser person. Because as Dr. Hamid and in the morning Dr. Zahir have mentioned, that don't think of challenges, think of opportunities. And those opportunities turn into our business opportunities as well. As we are working as my senior vice chairman Adnan was time and again was poking me that ultimately we all are commercial enterprise. We have to concern our bottom lines. Either we are working for a company or we own a company or we are MDs or chief executive of the companies. We are concerned about our bottom lines. But since we are into the human business, we are providing health care, kind of a thing, and we can't compromise on quality. Quality, efficacy. That should be our prime concern, and the economy comes itself. That I've learned through time and years. May I ask Professor Dr. Zahir to come in front so that Adnan Hirani Saab will present you a small token of our appreciation.